Hello lovelies, in this video the brilliant Dr. Edwards is going to be taking you through your AQA A-Level Biology Paper 3 predicted paper for 2023. Now I'm sure you are aware, but this is the paper that has the essays in it. So in this paper we're not only going to be taking you through the science, but there is a whole massive chunk at the end on how to plan the essays. So we're going to be showing you the planning bit that you should be doing and then an example essay. If you want more things that could be linked in, then there is the essay book over on my website. But this is a really good place to pick up lots and lots of revision tips, lots and lots of things you could add in as extra context and see how you can structure an essay in the exam. So if you haven't done the paper already, I would definitely recommend trying the paper before you watch this video, or you can just jump straight in, pause, try and answer the questions yourself, and then Dr. Edwards will take you through what sort of things the exams are looking for, how you should be structuring this answer, what you should be doing if the command word says this. Good luck guys, we're going to be here with you every single step of the way.
Hi everyone, welcome to our final A-Level AQA Biology Paper 3 walkthrough video. So it's the last paper out of the 3 papers. This is the one that contains the essay. So we've got the 2 sections, so the exam questions, which I'll go through step by step. I'll do the same as before. I'll give you the kind of tips on how to analyse them and figure out what they want in terms of content, talk about the answers, explain the mark schemes. And then I'll do the same for the essay. So for the essay, I'm going to go through both essay questions and I'm going to kind of show you how I plan the essay, show you a written kind of really high level marked version of the essay, talk about why it's going to get those good marks, and then go through kind of some of the, there's not really a mark scheme, there is a mark scheme for the essay, but there's not really kind of a mark scheme for each type of question. So we'll go through the general mark scheme, but then I'll also give you some like tips from examiners about the problems with answering those 2 essay questions in the past, and then that will likely give you some help with marking your own one at home. The Hawaiian Islands have the most diverse collection of drosophilid flies in the world, with approximately 800 different species having been identified. The species have been traced back to a single common ancestor from the mainland USA that colonized the island millions of years ago. It is hypothesized that this could have been a single fertilized female blown across in a storm or trapped in a piece of fruit stuck to a bird. Okay, so before we read the questions, this is already kind of pinging out to me that this is going to be a speciation question. We've got kind of geographical isolation, this idea of islands separating each other. We've got evolution because we've got kind of one single common ancestor and loads of species, kind of what we call adaptive radiation, so like radiating out from that one kind of common ancestor from the mainland, and then getting to over 800 different species. So that's kind of content knowledge we're going to need to answer this question. Okay, so part A says, what is a gene pool? Okay, so nice, easy sort of definition question to start with. Hopefully we know that the gene pool is just all the different alleles in a population for a gene. Part B, explain how the Hawaiian species of flies could have evolved. Okay, so this is our speciation question. So we need to include this idea of geographical isolation because we've been given those clues in the question. And then we're going to need to talk about obviously natural selection happening on the different islands which have different selection pressures because of their different environments, which is obviously going to trigger the different behaviours, and therefore the natural selection process on the islands will be different, which will favour different alleles. And then we get our kind of different reproductively isolated populations that are so different over a certain amount of time that they can no longer interbreed with each other, so they've become separate species. So this is kind of my bullet points that we're going to need to include, the steps that I would always try and include in this, taking those kind of bullet points and written them out maybe a bit more long form. So the flies are geographically isolated on the islands, so no interbreeding can occur. That's important. There is variation in each population due to mutation. That's really important. We have to mention this whenever we talk about natural selection or evolution. The environment on different islands produces different selection pressures. Here, that's really important because we're not going to go into massive detail about describing natural selection. We're just giving this idea that there's different selection pressures, and therefore natural selection will happen differently on the different environments or on the different islands. So then we say that natural selection occurs on the islands where certain alleles will allow organisms to survive and reproduce, changing the frequency of alleles in the population. And then obviously that is how we end up with a new species, or that, the new species has evolved that way because then eventually obviously they won't be able to interbreed with each other. On to part C. Drosophila are one group of insects which contain polytene chromosomes. These are extra large chromosomes found in the nuclei of salivary glands. They can be removed and stained, so their banding patterns can be viewed under a light microscope. Explain how scientists could have used the banding pattern on the chromosomes to establish the evolutionary relationships between flies. Okay, so hopefully we've seen pictures of chromosomes where you can see the kind of bands, the stripes on a chromosome. And obviously we know that that is representative of kind of the genes and the loci of the genes on the chromosome. And what it's really asking us here is, how would we compare that pattern, which is kind of like a barcode? How would we use that pattern, and how would we be able to tell evolutionary relationships from this? It's very similar to the questions where it would ask you, where you would give an answer to something like you could compare the DNA base sequence, or you could compare the amino acid sequence, or the RNA base sequence. And this is the same thing, we're just comparing the banding pattern. And then how would we link that to evolutionary relationship? Well, the more closely related you are, the fewer differences there are. Or the less closely related you are, the more differences there are. So that's basically what we're going to say for this question. That's what it kind of wants us to say, is kind of, how could you use this feature instead of, say, DNA or amino acid sequence? How could you use this to determine evolutionary relationships? And it's the same difference. The more differences there are, the less related, or the other way around. So they could compare the banding pattern on the same chromosomes. This is important because obviously it's about looking at differences in the same chromosomes for each species. So each species look at chromosome 1, each species look at chromosome 2, etc. Compare the banding pattern, and then the more differences there are, the less closely related those species are. About 100 species of the Hawaiian Drosophila have large, highly patterned wings and are known as the picture wing Drosophila. In some species, e.g. D. transipena, the males have more detailed patterning on their wings than the females. So suggest how the variety of wing patterns displayed by these flies helps to maintain them as separate species. Okay, so when we have deliberate, sort of really significant differences in the colour or patterning or behaviour of a male or a female, so one sex of a species, then that is down to sexual selection. And also this is linked to this idea that we have of like mating behaviour, courtship behaviour. Why is it really important that species are able to identify members of the opposite sex of the same species? Well, it does exactly this thing. It helps to maintain separate species because it stops you breeding with the wrong species and then interbreeding happens. So it makes sure you're only mating and successfully mating with a member of your own species to make sure that the gene pools don't get mixed up. So yeah, anything that helps make sure mates are of the same species, that helps a female or a male, depending on which one has the distinctive features, sort of identify a member of that opposite sex of the same species so they make sure that their breeding is going to be successful. So that's all we're saying here for 2 marks. So the pattern helps to recognise mates of the same species, and that stops us from interbreeding or mixing gene pools. Okay, so let's look at the mark scheme for question 1. So all the alleles in a population, that's just a definition that we should know. For gene pool, only 1 mark getting that right. For part B, we have to say geographical isolation, because obviously it's made it sort of clear in the question that this is the idea of islands, separating islands. So we're not talking about other types of speciation, we're talking about geographical isolation. If you talked about reproductively isolating, that's fine, or you talked about the fact that there's no interbreeding or there's no gene flow, because obviously we have to have separate populations where there isn't any interbreeding in order for this speciation to happen. We always say when we're talking about natural selection, whether it's to do with speciation or just a natural selection question, we always have to say variation is caused by mutation. Then, as I said, the idea of different selection pressures occurring in the different islands or the different habitats, or you could say different abiotic and biotic conditions or environments. That's fine, but this idea of different selection pressures due to the different environments in the habitats is what's going to cause and drive these different natural selection processes that are going to happen, which are going to make our different species. And then we've got sort of a brief statement on natural selection. So different reproductive successes in selective organisms, or certain organisms survive and reproduce, or certain organisms will be able to pass on alleles or genes to the next generation, something along those lines. And it needs to be sort of a brief explanation of how we get different organisms with different characteristics surviving and reproducing, which is obviously natural selection. And then it leads to change or an increase in allele frequency. So obviously those organisms that have those characteristics, those alleles that are going to help them to survive and reproduce in each of the different habitats with the different selection pressures, they're the alleles that are going to get passed on to the offspring. So those alleles increase in frequency in the population until we get different populations that cannot interbreed with each other, which is what speciation is. So part C, as we said, so compare the banding patterns. That's the main thing on chromosomes from different species or the same chromosomes, but realistically it's about talking about comparing those patterns. And then fewer differences or more similarities, they're more closely related or they're more likely to share a common ancestor, or you can flip it and say the other way around as I did. Therefore, as we said, so it's kind of like mate selection and importance of making sure that you're mating with someone of the same species as you. So wing pattern is used to determine mate choice, or as we said, like determine selecting a mate of the same species. And it keeps gene pools separate, or it prevents interbreeding. You can't just repeat the question here, so you have to say one of these two for the second mark to make it clear how you're keeping the species separate. So preventing interbreeding between species, or keeping gene pools separate and making sure there's no mixing. Some organisms are able to fix atmospheric nitrogen using a nitrogenase enzyme. Nitrogenase enzymes can be inhibited by high oxygen concentrations. Cyanobacteria are one of the few organisms that photosynthesize but also fix nitrogen. Question A says, explain how each of the following adaptations allow cyanobacteria to carry out both processes. Okay, so the information we've been given here might be something we don't know, but let's sort of just make a few notes to recap it. So high oxygen stops nitrogenase enzymes from working, so it stops nitrogen fixing. Obviously, if you're going to carry out photosynthesis, you're going to be producing oxygen in your cells, and then that's going to be inhibiting your nitrogenase enzyme. So we need to explain how these adaptations allow cyanobacteria, which do both, so they photosynthesize and they fix nitrogen. How does it allow them to do that? Well, if they're going to do both, they must do it in a way that would stop oxygen being produced or stop oxygen being present in cells where the cyanobacteria are also doing nitrogen fixing. So the first adaptation is that some colonies of cyanobacteria develop heterocysts, which are specialised cells which only contain photosystem 1. Okay, so this is an adaptation to how they do photosynthesis. So if they're only using photosystem 1, the product of oxygen during photosynthesis comes from photolysis. If you only have photosystem 1, no photolysis is going to be taking place, because if you remember rightly, only photosystem 2 has the enzymes available to carry out photolysis. Photolysis only happens at photosystem 2. So if you only have photosystem 1, that means you can still be absorbing light, you can still be exciting electrons, you can still have electron transport chain, but you're not doing photolysis. So therefore there's no oxygen being produced, so then therefore we're not inhibiting the nitrogenase enzyme. Our second one is that some only fix nitrogen at night. Okay, so kind of similar answer here, because if there's no light, then we have no light-dependent reactions of photosynthesis happening, which is where photolysis occurs, because we need light to split water. That's what photolysis is. So therefore, there's no oxygen being produced, and therefore, we're not inhibiting our nitrogenase enzyme. So we're kind of going to write the same thing, except we're just going to write there's no light at night, therefore no light-dependent reactions, so no oxygen to inhibit nitrogenase enzyme. Some plants make associations with nitrogen-fixing bacteria in specialised structures known as root nodules. Genes are activated during root nodule development, which code for the protein leg hemoglobin. This is a type of hemoglobin that is very similar to animal hemoglobin. It also binds oxygen. Okay, so I've put the paragraph from earlier in because we are going to need this, because that was about nitrogen fixing, and this question is about nitrogen fixing as well. We're just talking about bacteria this time. Leg hemoglobin is potentially something we've never heard of before, but it's telling us that it's similar to animal hemoglobin, which means it still has a heme group, and it also binds to oxygen. So we're going to need to think about that when we read the question. To investigate this action in nodules, scientists used siRNA to silence the leg hemoglobin genes. So they stopped the expression of the gene that codes for the leg hemoglobin protein. So that means there'd be no leg hemoglobin being produced, which means it won't be there to bind to oxygen because it says it binds to oxygen. That means that the oxygen that would normally bind to it will be in the cells or in the tissues or in the nodules, which means there'll be an increased concentration of oxygen than normal, which means we're going to have a decrease in the action of the nitrogenase enzymes, which means a decrease in nitrogen fixation because those enzymes will be being inhibited by the oxygen concentration. So there we go. That is everything that we can apply our knowledge to or get from all of that information at the start of the question. The scientists observed that these modified plants exhibited less than normal growth. Only after addition of nitrogen fertilizer were the plants able to grow normally. Suggest why. So because they aren't able to do nitrogen fixation because of these reasons that we've said, they need nitrates from the soil in order to boost their growth. Because they aren't getting enough nitrogen from nitrogen fixation. What do plants need nitrates for? Well, they need nitrates in order to be able to make amino acids, to make proteins, in order to make DNA. So that's where this kind of link to growth then comes. So although this is only 2 marks, and I feel like it's obviously seems like it's a lot of information for 2 marks, but if you think about it, we got all of that first part just from reading the question. And then we're just really getting marks here for linking lack of nitrogen fixation to lack of growth by talking about the fact that they're not going to be able to make DNA or amino acids, whatever we choose in terms of molecules in order to grow. So the leg hemoglobin was not present to bind to oxygen, so will increase and inhibit nitrogenase enzymes. The plants do not get or do not grow because they're not getting enough nitrogen in order to make amino acids, or you can say DNA, whichever you choose. Taxol is a drug used to treat cancer. It was extracted from the bark of the Pacific yew tree. Scientists initially tested this drug on mice that had visible cancerous tumours in their lungs. Some results are shown in the table below. So we've got the day of how many days they've been treated, we've got some control mice, and we've got some mice injected with Taxol, which is the drug in saline, and we've got the mean volume of the tumour in millimetres cubed. Suggest and explain 2 factors which should be considered when deciding on the number of mice to use in a trial such as this. Okay, so whenever we're thinking about trials involving animals, or humans, but sort of this is more to do with animal trials, especially obviously mice are really commonly used for things like this, or rats. We need to consider the ethical implications of the fact that obviously we know and understand we need to use animals in testing of drugs, but we don't want to cause undue suffering to lots of animals if we don't have to, because they are going to suffer and get cancer and probably die, especially the control mice who are not going to be receiving any treatment. The sample size, you need a lot, but you need to balance that, obviously, with a large enough sample size to make sure that your data is reliable and that you can draw conclusions from it that are valid. And the cost and upkeep of these animals as well. So the more animals you have, the more it's going to cost to look after them, feed them, house them. They're going to need people to look after them, so that requires maybe hiring people to look after them, cleaning the cages out, and ethically looking after them in a good way, which is obviously to do with cost, upkeep, space, and obviously that's dependent on how much cost, sort of budget you have, how much space you have. So these are sort of all the things that we've got as options here to think about when we're thinking about how we look after or what we consider when we're doing animal trials. So I'm going to choose the ethical considerations, as mice are likely to suffer or die, and die, and the cost involved in having a large number of mice.
Part B.
The scientists concluded that Taxol did not kill cancer cells. Does the table support this conclusion? Explain your answer. Okay, so we need to say yes or no, and then we need to back up our answer using evidence, reasons from the table. And it's asking us if the Taxol did or did not kill the cancer cells. We obviously don't have that as a measure. What we have as a measure is the volume of tumour. And obviously the tumour is made up of cancer cells, and cancer cells dividing rapidly would cause the tumour to grow. If the cells were dying, or if the cells died, then the tumour would get smaller because the cells would die. We'd expect the tumour to shrink or decrease in size. So we need evidence from the table as to whether the tumour is shrinking or not shrinking. And if it's not shrinking or not decreasing in size, then we can support this conclusion that it's not killing the cancer cells. But if it is shrinking in size, then that would not support this idea that the cancer cells aren't dying. So let's have a look at the numbers in the table. So you can obviously see that in each row, on each days of treatment, 10 days of treatment, we're having an increase in number. So clearly, it's still increasing. Although the Taxol treated mice, there's not as much of a rapid increase in growth, so it's slower growing, but it's still growing. There's still an increase with every single day. There's not a levelling off. There's not kind of a preventative or a decrease in the size or the volume of the tumour. So we can say, yes, this table supports the conclusion that the Taxol is not killing the cancer cells because the tumour is not shrinking or decreasing in size. It's keeping increasing, just at a slower rate.
Part C. Taxol works by disrupting spindle activity. Explain how Taxol affects cancer cells. Okay, so the spindle fibers, the spindle activity, they obviously get produced and they attach to the chromosomes at the centromere, and they move the chromosomes around during mitosis. And so if that isn't happening, if that's not able, or it's disrupted, or it's slowed down, then cell division itself will slow down. And we know that, and we've seen that from the evidence in the table where it obviously said that tumour growth was slowed down. So we need to say that cell division slows down, or the cell cycle slows down, but we can't say it stops because we know that it doesn't stop growth because we saw that as part of the evidence in the table where we said that it was still growing. So we need 3 things for 3 marks here. So talking about the chromosomes not being able to attach to the spindle, talking about the fact that mitosis itself, that happens during mitosis and that will be affected, and then the 3rd mark for talking about cell division. Part D.
A more targeted drug needs to be developed. Aurora A is an enzyme that phosphorylates the protein TAC3. So TAC3 is the substrate that obviously binds to the enzyme. And TAC3 then binds to microtubules, which hold spindle fibers together. Aurora A is found to be amplified in tumour cells. That makes sense if it's to do with sort of binding microtubules and holding spindle fibers together. More spindle fibers are being created and manipulated because more cell division is happening, and obviously we need spindle fibers to do that. So it wants us to suggest how scientists could develop a cancer drug which targets Aurora A. So that means it targets that enzyme specifically and stops it or reduces it, stops it working, or inhibits it in some way, slows it down, reduces its effect. Anything to do with that as a way of explaining how the drug could work. It's a suggest question, so it doesn't mean there's one specific right answer, but using your knowledge. So I would say if you're being asked of a way you could think of to prevent an enzyme from working, the easiest thing to think about is either competitive or non-competitive inhibition of that enzyme. So that's something that we've learned in the course, so that we're expected to be able to know that, and it's something that we can hopefully easily describe for 2 marks. Okay, so if we're going to say competitive inhibition, as I'm going to pick for my answer, then we need to say how it would work. So for 2 marks, so I'm going to say, obviously, it's a similar shape. So the drug will need to be a similar shape to the substrate. In this case, it's TAC3. So it would need to be a similar shape to TAC3. So it's going to compete with TAC3 for the binding site, for the active site of the enzyme. And that's enough. That's all we need to say. So we're making the drug with a similar shape to TAC3. And what's that going to do? How would it work? Well, it's going to compete with TAC3 to bind with the active site of Aurora A. And obviously that would hopefully reduce its functionality. So there's our 2 points. And we'll have a look at the mark scheme to see what other options you could have put as well.
Okay, so the mark scheme for question 3, we've got the ethical consideration, the number of mines being related to cost. So as we said, these are kind of typical questions you might get or might think about as considerations for when you're doing trials that involve animals. So anything to do with ethics is always obviously important, especially if you're doing things where they're sick animals or you're making animals sick and then you're treating them with a drug that could be dangerous, you don't know, but also obviously the control organisms will suffer and likely die because they're not getting any treatment. So you don't want too many animals to suffer in this way. Then obviously you've got to think about having a large enough sample size. We didn't really talk about this, but making sure you've got enough, sort of balancing that, not wanting to have too many, but having enough to make sure that any data you're getting is really valid and reliable. And then the other thing that's associated to like the number is that it might be limited by how much space you have available, how much budget or time you have to be able to make sure all the animals are cared for, fed, watered, their cages cleaned out and things like that. All of that's going to depend on the budget and how much kind of time, manpower you have. And so any 2 of those got you those marks, but you needed to obviously say, couldn't just say ethics, you need to link to it. Couldn't just say large number, you need to say why. It's an explanation. Okay, then part B, yes, and obviously we don't get a mark for saying yes. We have to say obviously because there's an increase there, it keeps increasing, or you can say it's getting larger or the volume is increasing. You can say at a lower rate, obviously it is slower, but that really isn't necessarily backing up the answer here because the answer would need to explain that why that shows that it's not being killed, the cells aren't being killed, and it's because obviously if the cells are being killed, it would decrease in size, it would shrink, there'd be fewer cells. So we have to say both parts here to get that mark. Then part C, as we said, we've got 3 marks. You have to talk about mitosis. Obviously this is disrupting mitosis as the part of the cell cycle that's being affected because of the spindle fibres. The spindle fibres obviously would normally attach to chromosomes and move them around, which we can't do, and then therefore cell division or the cell cycle slows down. It says obviously if you just said mitosis is stopped, that's not enough. If you just said mitosis slows down, you get you 2 marks, and then you would need to say the why and then talk about the obviously chromosomes not being able to attach. And then part D, the important thing here is obviously there's options for what you can talk about. So the answer here is for competitive inhibitor. If you talked about a non-competitive inhibitor, you can also get marks here. So if you talked about the fact that it binds away from the active site and then it causes a change in the shape to the active site so it can no longer bind, that would get you marks, obviously standard marks for that type of answer. If you went for competitive inhibition, then you needed to say similar shape or similar tertiary structure. It can't be the same. Or you can say create a drug with a complementary shape to the active site. That's also saying the same thing there. It's complementary. It doesn't say it's exactly the same. And then you need to say obviously it would compete with TAC3 for the active site, or it would bind to and it would block the active site of Aurora A. There's no marks here for talking about gene silencing or preventing transcription or translation of the gene that codes for Aurora A, because that's not in the question. The question asked how would you target the enzyme specifically. So just need to be careful when reading the question, especially when it's these kind of suggest questions, that we don't kind of go off and talk about something that's not related to the question.
RT-PCR, or reverse transcriptase PCR, is used to test for RNA viral infection in patients. So reverse transcriptase, I'm making a note before I forget, so it's the enzyme that turns RNA into DNA. So it normally turns mRNA into cDNA. Obviously, it's something that viruses possess. It's an enzyme that they have that allows them to obviously inject RNA, but then convert it to DNA so that then the cell's machinery will use it and replicate it. It can accurately measure the amount of RNA in a sample. The RT-PCR uses the same reaction mixture as a standard PCR, except that reverse transcriptase enzymes and a fluorescent dye that binds to DNA are included. Okay, so we've got some information about a type of PCR that we don't know, but we've been told that it's pretty much the same as normal PCR.
So part A,
state 2 other ingredients which must be present in the RT-PCR reaction mixture. So that's saying basically what ingredients do you know has to go in the PCR mixture. So hopefully we know that we need a template strand. In this case, obviously, it's going to be the RNA. It's going to be the sample RNA. So you can't say DNA in this occasion. We're going to need DNA nucleotides. We're going to need primers, forward and reverse primers. And what else are we going to need? We're going to need DNA polymerase as well. It's really important with these types of questions that you make sure you know whether you're talking about RNA or DNA. And if you're talking about nucleotides and you want DNA nucleotides, put DNA in front. If you're talking about polymerase and you talk about DNA polymerase, put DNA polymerase in front. Because you are going to be likely to lose marks if you don't say the type of nucleotide or polymerase here. Because obviously we know that you could be meaning RNA polymerase. If you didn't understand what reverse transcriptase did, you could get this wrong. So DNA nucleotides and DNA polymerase is what I'm going for. You can go for any two off that list. Now it wants us to explain the role of reverse transcriptase in the RT-PCR. Well, we've already said that. It's basically a question just asking us, what does reverse transcriptase do? So it converts RNA into cDNA. Next part. So it says RT-PCR can be used to test if a patient is infected with a virus. A test is considered positive when the fluorescent signal reaches a certain threshold. The CT value is the number of PCR cycles required for the fluorescent signal to cross a certain threshold. RT-PCR was used to test if patients were positive for the influenza virus. The result of one of the RT-PCRs are shown in the graph below. So we've got the fluorescence intensity. So the more fluorescent it is, clearly there's a dye that binds to DNA, so the more DNA there's been produced from the replication cycles. Then we have the threshold level. So this is the CT value is going to be the point at which this sort of line of DNA and the threshold value meet. That's going to be the number of cycles. So it takes 30 cycles here for the CT value to be reached, for enough DNA to be reached that it is detected. So that would be saying, okay, it takes 30 cycles to get enough DNA or RNA present in a sample converted to DNA that then is fluorescing enough to be considered present. Suggest one explanation for the levelling out of the curve from cycle 50 onwards. So we should hopefully know and be able to explain why PCR graphs level off like this. So they have this S shape. The slow start is because obviously there's very few amount of DNA to start with and it replicates. It needs to get to that exponential level before we can start seeing a big increase. But the sort of levelling off at the top is due to the primers running out most often. There's some other things we can talk about and use in our explanation, but that's what I would normally say is the reason why we get a levelling off. If the primers are running out, we need to explain why that means a levelling off because it's 2 marks. So just saying primers running out is not going to get us the marks here. We need to explain. Well, the primers obviously bind at the start of the sequence and allow DNA polymerase to bind, and then that's what starts a new strand. No new copies are going to be started because there's no more primers there to bind, which means that DNA polymerase won't bind, so that's why we're not going to get new strands. So the primers may have been used up, so no new copies or no new strands can be started. How many cDNA molecules have been produced from one molecule of DNA at the threshold value of the sample? Your answer should be in standard form. Okay, so for this, including like bacterial growth curves, there's like a sort of quick cheat maths way of doing sort of doubling time. So figuring out how, if something's just doubling, how many there'll be at the end of a certain point. You just do the number at the start. So in this case, it'll be 1 because it's just a sample, times 2 to the power of, in this case, the number of cycles, because the number of cycles equals the number of divisions. If it was a bacterial growth curve, you just do the number of times it divided in that amount of time. So it wants us to do the 30 because that's the threshold value, and we read that off the graph earlier. So
1 times 2 to the power of 30 is a very big number. 1,073,741,824. But it wants our answer in standard form, remember, so 1.07 times 10, and if you want to make sure you get the number of the power correctly, do the little jump method. So take it from where the decimal point is to the end, that is 9 jumps, so it's 1.07 times 10 to the power 9. Because RNA degrades slowly over time, it may still be detected many weeks after a patient is no longer infectious. In order to confirm if the RT-PCR was detecting RNA from the active influenza virus, the lab would also carry out a viral culture test on the same sample. This method is detailed below. So we culture animal epithelial cells were inoculated with 500 microliters of solution, containing 33 microliters of the patient's sample and 2 times penicillin streptomycin solution. So the first step is to basically infect some animal cells, or if there is live virus there, they will infect these animal cells. If they take a patient sample, if there's viral there, it's kind of actively replicating. So if it's not latent, if it's actively replicating, it should infect these epithelial cells. And we're adding penicillin streptomycin solution, which is an antibiotic solution. So this is obviously going to be here to kill any bacteria. So we're obviously infecting with the virus, and we've got animal cells, so neither of those will be affected by the antibiotic. The cells are maintained in a 37 degrees C incubator in human plasma-like medium. Okay, so we're clearly replicating the body conditions here. We've got body temperature, we've got human plasma-like, so surrounding them in basically tissue fluid to really replicate what it would normally be like in the body. Samples of cells are removed daily for observations to identify cytopathic effect. Okay, if we don't know what that means, that's fine. We'll probably come back to it. There'll be something in the question. So part E says, suggest why penicillin and streptomycin were added to the cell culture. So why are we adding these antibiotics? As we said, they're going to kill bacteria. That's still not why. So that's sort of what they do, what they are. Why we added them is because they're going to kill any bacteria that could cause contamination. We're obviously growing stuff in a situation where if there was bacteria present, or if it was accidentally contaminated with bacteria, then there's a potential it could contaminate the sample by sort of replicating. So it's to kill bacteria and prevent contamination. And then finally, part F. Cytopathic effects refers to physical changes in host cells caused by attacking viruses. Okay, so that's what that means. When viewed under the microscope, suggests what visible effect viral infection would have had on the cells. So it's basically asking, what if cells are infected with a virus, what will happen to those cells? What could you see physically happening to those cells? What would a virus do? We've already had part of the question of this question. It feels like a long time ago now because it's about 6 parts ago, but in the start of the question they talked about enzymes being present that are able to break open the cell membrane. And we know that that's how viruses replicate. So they replicate inside the cell and then they will burst out, or they have to get inside the cell somehow. And we know from earlier on the question that they're going to break open the cell membrane. So we're expecting we'd see damaged cells that have damaged cell membranes, broken, damaged cell membranes, or lysed cells, which just means like burst cells. And so that's what we'd be looking for. If viruses were coming through and infecting and damaging and killing these cells, that's what we'd see under the microscope.
Obviously, as we said, if you're going to say what's going to be in the reaction mixture for the PCR for part A, you need to say DNA polymerase or DNA nucleotides. You can't just say polymerase and nucleotides, you need to be specific because obviously you could be talking about RNA. And then we've got primers, and then you've got your sample RNA or your test sample. So you just need 2 of those correct to get 2 marks. The kind of action of the enzyme, you were allowed converts mRNA to cDNA or RNA to cDNA is fine, or just RNA to DNA, sorry, because the C and the M are in brackets. I would try and make sure you do say cDNA. In this case, you didn't need to say mRNA, but I would try and say cDNA because you are obviously making complementary DNA from mRNA. You're not making what we would call normal DNA. And then you can have 1 suggestion with explanation for the 2 marks for part C. So, as I said, most likely it's going to be that the primers are being used up, so you can't start new chains being copied. You can put this, and it's not wrong, nucleotides being used up, it's just very unlikely. So obviously you've got less, so you've got nothing to make the new chains, which is your explanation for that point. Or because we are obviously heating, cooling, enzymes could lose their activity or denatured, so there's no polymerization of the complementary strands, because obviously the only enzyme we have is DNA polymerase. But I would say, if you see this question in the exam, because it has come up, I would say try and go for primers. Then if you can't think of primers, nucleotides your second choice, but it is less likely. We've got obviously our CT value was 30 off the graph, which we obviously read off, which is where the line crosses the threshold. And then you needed to work that out using our kind of standard method for figuring out doubling. So 1 times 2 to the power of the number of doubles, so that's 30. And we've got a very, very big 1.07 billion. So just make sure you get that in standard form, and you get 2 marks for this, so you get 1 mark if it wasn't in standard form. To prevent the contamination with bacteria is kind of the main reason why you treat anything when you're growing cells in a lab with antibiotics. So that's the kind of easy way. You can say it in different ways. So to prevent bacterial infection, so that any effect would be due to the virus and due to a bacterial infection. So that's kind of another reason, sort of a reason why we don't want bacterial infection. We obviously don't want those cells to be damaged in any other way than the virus that we're infecting them with, because otherwise how would you know to look out for the virus? And then lastly, this is just, as we said, a cell, a sort of what happens to a cell if it's infected with a virus question. And so you can say lysed, which is just obviously the fancy way of saying burst cells, or ruptured or broken or damaged cell membranes. On to question 5. Diabetic retinopathy is caused by persistent high blood sugar levels, damaging blood vessels that feed the retina. The vessels can become weakened and they can leak, causing bleeding into the space just under the retina. If this occurs near the fovea, a fibrous scar is formed that destroys the photoreceptor cells, reducing visual acuity, and will eventually lead to blindness. Okay, so this is a very typical paper 3 question, where it's going to be all about something that we really haven't got much detail on, or we don't know from the course. It's talking about diabetic retinopathy, it's talking about bleeding into the retina, it's talking about the fovea and forming a scar that destroys photoreceptor cells in the fovea. So hopefully, and it reduces visual acuity. We hopefully know what visual acuity is because it produces detail, and we've heard of the fovea because that's a dense kind of area where there are cones, and obviously cones are responsible for this visual acuity and detail. And so that's where our content is coming from for this question to start with. And realistically, that's where we need to be thinking, ignoring the other information for now. So part A says, explain why visual acuity is affected if bleeding occurs near the fovea. So we've been given this information about the fact that obviously we can have bleeding by this area of the retina called the fovea. We should know that there's lots of cones there. And so obviously that's our knowledge there coming from that, and also knowing that cones are related to visual acuity. So we need to say why visual acuity is affected. We can't just say because there's lots of cones there, because it is for 2 marks. So we need to link the actual reason why visual acuity is linked to cones. So we have to talk about the fact that cones are bound to only 1 bipolar neuron each, or each cone is attached to only a single neuron. So the signals from each cone are sent separately. There is no summation of signals. They're not joined to the same bipolar neuron. So therefore, that's what allows us that detail. So our first mark is going to come from saying that there's a lot more cones in the fovea, which have separate bipolar neurons. So there is no summation of impulses, or impulses are sent separately. Part B. So this is just a maths question, which again they can throw in anywhere. So the circular fovea in a human eye has a diameter of 3 times 10 to the power of 3 micrometers. An area at the centre of the fovea called the foveola has a diameter of 300 micrometers and contains no rod cells. There are approximately 1.7 million rod cells in the fovea. Estimate the density of rod cells per millimeter squared in the fovea. The area of the circle is pi r, or calculating the area of the circle is pi r squared. Use pi equals 2.14 in your calculation to show you're working. Okay, so I've done like a little sketch just so I can really visualize what that information has been given to me. So the diameter of the fovea is 3 times 10 to the 3 micrometers, And then the foveola is obviously a circle or an area in the centre, and that is where obviously the density, or there's a dense patch in there where there are no rod cells. And so that diameter is 300 micrometers. So we need to find out how the density of rod cells per millimetre squared in the fovea. So there's only rods in the fovea, not in the foveola. So we've kind of got this donut space where the rods are going to be. So in order to work out the density in the area, we're going to need to work out the area of the small circle, take that away from the area of the large circle to find out the area we have left, and then divide our 1.7 million by that area. But that area needs to be in millimetres squared, not micrometers squared. So we're going to need to convert that area once we've got it into millimetres. And obviously we're going to need to use pi r squared. So obviously we're going to need to use a calculator for this. And obviously remember we've been given diameters, not radius. So we use a half the diameter to convert to radius in order to make sure that we're using the right numbers. So we're going to do 3.14 times 1.5, which is the radius of the fovea, and square it. And then 3.14 times 0.15, which is the radius of the foveola, and square it. Take the small area away from the large area, and that gives us 6.99435 millimeters squared. So now we're going to have to think of what we're going to do with that number. So we're going to take our 1.7 million and we're going to divide it by that number, which then will get us the number of rods per millimeter squared. So that gives us 243,053.32. If you're going to round that up, and you can put it in standard form as well, so it's 2.43 times 10 to the 5 millimeters, so per millimeter squared. So we write that as millimeters minus to the power of minus 2. So there we go. That is the density of rod cells per millimeter squared in the fovea. Part C. One treatment for retinopathy is to inject medication directly into the eye. To do this, local anaesthetic is given and eye drops are used to keep pupils dilated. The eye drops contain a synthetic form of atropine, which is a toxin extracted from plants. Atropine blocks acetylcholine receptors, preventing depolarization. So yeah, if you've ever known anyone who's ever had any kind of eye surgery or eye work done, then they do give you these drops that make your pupils dilate and get really large, because obviously that's the hole that they are able to then go into the eye and get lasers or whatever is needed to get through that pupil to the back of the eye. So they get the more vision. If your pupil is really large, they've got more space to work with. So this atropine, whether you've heard of it or not, it tells us it blocks acetylcholine receptors, preventing depolarization, which means it must have a similar shape to acetylcholines that can bind to those receptors. That's what it's making me think of straight away, even before we've read what the question is. These iris muscles are under the control of the autonomic nervous system, which deals with involuntary reflex actions. Which branch of the autonomic nervous system is inhibited by the eye drops? So this is a question about whether we are going to be affecting the sympathetic or the parasympathetic nervous system, as these are the two branches of the autonomic nervous system. We need to know that. Obviously, when we focus on this, obviously we're thinking about control of heart rate. So this is kind of applying that knowledge to something different. You'll notice this is like a massive sort of synoptic question. So we've gone from talking about the eye and kind of how the eye works to a mass question to now talking about the sympathetic and parasympathetic nervous system. And we're going to do this. The next part is about muscle contraction, and then we're going to go back to doing some data analysis. So really, really big synoptic question here with loads of different parts. So really this question is asking, to dilate the pupils, are muscles contracted, which would be the sympathetic nervous system, or are they relaxed, which would be the parasympathetic nervous system, because one is about sort of activating and one is about relaxing. So in order for your pupils to be dilated, the muscles around the iris need to be relaxed. And that is what's going to cause them to the pupil to open wider. So clearly we're going to be affecting the parasympathetic nervous system here. Part 2. Explain how preventing depolarization stops the muscles contracting. So this is a clue. If you didn't know about the relaxing of the eye muscles to in the pupil, then this tells you it here, because we're preventing depolarization, we're stopping the muscles from contracting, which must mean that we're making them relax. So that is why a clue to that previous question, which is helpful. It wants reasons why stopping depolarization would stop muscle contraction. So we need to link depolarization to muscle contraction. So what does depolarization normally cause that leads to muscle contraction that won't happen here? So a lot of these biology questions, when they're asking us sort of short questions about processes that are quite complicated, are going to be like, well, what happens if this doesn't happen? What happens if this doesn't work? What happens if we stop this from happening? So you have to know your processes, be able to flip them backwards and say, okay, well, if that doesn't happen, then that won't happen. So in muscle contraction, depolarization causes the release of the calcium ions, and the calcium ions then obviously bind to tropomyosin, move it, and expose the binding sites on the actin, which allow the myosin heads to bind. So if we don't have depolarization, because it says that, so we're going to say no influx of calcium ions, so the binding sites on the actin are not exposed, so the myosin can't bind to the actin, and that's why we don't get muscle contraction. So this is only a 2-mark question, so we don't have to write loads of detail here, but we're going to obviously get 1 mark for saying stopping the influx of calcium ions, and then our second point is obviously saying so the tropomyosin isn't moved, so the binding sites on the actin are not exposed or are not free to be bound to. Many eye diseases that cause blindness, including diabetic retinopathy, are due to the overproduction of a protein called vascular endothelial growth factor, or VEGF. VEGF is essential for the growth of blood vessels and normal growth and development of many other organs, including the lungs, kidney, and brain. But overproduction of VEGF can be triggered in tissues that are not receiving enough oxygen. Okay, interesting, might be worth remembering that. In the presence of excessive VEGF, more blood vessels become leaky, and new vessels that grow are also vulnerable to be leaky. So obviously blood vessels leaky, meaning that blood and other fluids can come out of them. That's obviously not ideal. Obviously there's a link here with not getting enough oxygen, so that might come up in a question. So we might need to talk about or link this idea of not enough oxygen and why VEGF is then triggered or stimulated to be produced. And then we've also got this idea of what VEGF does. So it obviously is helping to cause blood vessels to grow, and that's obviously important when you're growing new tissues and clearly organs that have lots of blood vessels inside them as well, like the lungs, kidney, and brain. So we'll keep all this in mind as we read the questions. One method of treatment is to inject anti-VEGF drugs. These are monoclonal antibodies which bind to all forms of the VEGF protein. Use your knowledge of monoclonal antibodies to suggest how these drugs work. Okay? Again, with the synoptic questions, we've now got a monoclonal antibody question, which is again not, related to any of the other topics in this question so far. So again, you just take sort of each question as it comes, make sure you're kind of thinking about the information they're giving you at the start of these questions and how you're going to use it. So it's a growth factor. It says VEGF is a growth factor. So that means kind of like a transcription factor. It's going to bind to something, a receptor that's going to trigger this production of blood vessels growing. So this makes sense because if we looked at and thinking about our content around monoclonal antibodies and how they've been used as drugs or to target various proteins, they obviously, the structure of antibodies is designed so that they can have that variable region that has a specific tertiary structure that can be complementary and bind to various things. So in this case, it could bind to VEGF. And then if the VEGF has all these antibodies bound to it, it's not going to be able to bind to any receptors in order to trigger the growth of blood vessels, which is what it does. So this is basically it for 3 marks. So we're basically saying what we understand about antibody structure. So the fact that it's got a variable region, the fact that it's got a tertiary structure that's specific, that can be made to be complementary to a protein that you want to target. So in this case, VEGF. And then we need the how the drug works. How does it stop VEGF? Because it's anti-VEGF drug. So the way it works to stop it is that it can bind to the VEGF and then prevent it binding to its receptor and stop it from carrying out its processes. So language is really important in our answer here. So the antibodies have specific tertiary structure, important, which is complementary, important, to the VEGF protein. And then we need the how. So that's our kind of what we know about antibodies. And then the how is it's going to bind to the VEGF protein and prevent it from binding to receptors. Three main anti-VEGF drugs are currently available to treat retinopathy. Scientists recently conducted a randomized blind clinical trial of all three treatments. They compared their effectiveness at improving visual acuity in diabetic retinopathy patients with medium to high vision loss. There were 660 patients in total. Okay, so that's immediately I'm going, okay, that's quite a high sample size, which I think is quite good. We can be quite confident about that. Patients had to be over 18 years of age with type 1 or type 2 diabetes, and clearly they have got diabetic retinopathy because it said that they were patients. So that's kind of quite a wide demographic, although it's obviously likely that there'll be, a lot of people in there with a similar kind of age range, because normally this is going to take place when you're an older person who's lived with diabetes. Patients were grouped according to the extent of their vision loss into high or medium category based on their visual acuity scores. Okay, one eye of each patient was randomly assigned the anti-VEGF, one of the anti-VEGF drugs, so Lucentis, Eylea, or Avastin. So if they're only using one eye, I think the main thing I'm sort of getting here is that if only one eye is used, then the other one can be used as a control. Other eye of each patient is used as a control. Some ethical issues there with why you'd only treat one eye. I guess because if there's a risk there, then the other eye is fine, but a bit suspicious. Treatment occurred every 4 weeks with injections for a year, and then every 4 to 16 weeks during the second year, depending on the recommended course of treatment for each drug. At each visit, the patient's visual acuity was checked using a standard Snellen chart of letters held 6 meters away. So that's the same as the standard eye test that you would go for. Every time you have an eye test, and you have to read those letters off the wall. Okay, so that's a lot of information here. I'm probably going to have to come back to some of this. We've got two really kind of big, long, complex graphs, one for medium vision and one for the high vision loss group. We're not going to look into those graphs right now because this question doesn't rely on it. We'll do that on the next page. So part E says, suggest why no pregnant women were allowed to take part in the trial. Okay, so obviously this trial is with anti-VEGF drugs. We've literally just answered a question that sort of tells you how that works, and it blocks the VEGF protein from working. And if you remember back at the start, or a few sort of pages ago, we talked about what the VEGF protein does. It stimulates growth of blood vessels. It said it was really important for the normal growth and development of blood vessels, but also certain organs, including the kidney and the lungs and the liver. So if you are pregnant, you are growing a fetus. You are growing a new human. And so that growing fetus is going to need VEGF protein to work and function, because it's important for the growth of blood vessels and also all those organs that we mentioned. So you can't take anti-VEGF drugs if you're pregnant in this trial, because those antibodies, especially if they are that type, will be able to get into the blood, and then they could cross the placenta and prevent VEGF from doing its job in the fetus, which is obviously helping it to grow and develop. So we're going to obviously say that the anti-VEGF drugs could affect the growth and development of the fetus, which is obviously not what we want. So that's why they were excluded from the trial. This is one of those ones where it's a suggest why question. So you had to use your knowledge of what being pregnant is and growing, but also basically just pick out information from the earlier part of the question that told you what VEGF does, and obviously make that connection that we don't want to stop that working in a growing baby. Part F. A student analysed the graphs and concluded that Eylea was the most effective and should be used in the majority of retinopathy cases. Evaluate the student's conclusion. Okay, so there's a lot of keywords in this question that are important here. So the idea that it's going to be the best or the most effective, is that true? And should it be used in the majority of cases? So in this case, we know they only looked at medium and high vision loss. So is it, in this case, they're asking, is it suitable for both high and medium vision loss? Is it the most effective? So is it better? Was the visual acuity scores improved the most with that drug consistently? Can we say that? And can we say that the effectiveness was the same for both the medium and the high vision loss groups? Because if we can't, then we can't support this conclusion. It's asking us to evaluate the student's conclusion. That means we need a yes or a no and why. So we can say, this is true, but this is not true, and therefore this is not the case for the drug. So we just need to be able to analyze the graphs ourselves to see and look for that pattern. So in graph 2, the high vision loss group, I'm going to this one first because it's easier to spot this pattern here. There is a clear distance where there is no overlap between the error bars on every single one of those points for Eylea versus the other two drugs. So at all points throughout the whole 2-year trial, there is no overlap. So there is what I would suggest a significant difference suggested that Eylea is the best out of those three. If we then go to group 1, which is the medium vision loss, we've got overlaps in 3 sort of almost 4 places across the time, mostly towards the end of the treatment though, so after about 84 weeks. So in terms of, although it seems initially to be more effective up until sort of 40 weeks or so, and there is no overlap, long term, there is not much difference, or maybe potentially not a significant difference in visual acuity scores at the end of the treatment compared to the Lucentis, which is the second, where they are overlapping the most. Yes, it's more effective than the Avastin, but there's not much difference there long term for the medium vision loss patient groups. We don't see that same sustained effectiveness that we see with the high vision loss group with the medium vision loss group. So that's what we're going to basically say. So we can say we've seen that no error bar overlap. We can say we've seen the overlap on graph 1. And so we can say that actually their conclusion is not valid because it is sort of the most effective, especially in high vision loss patients. It's not going to be the most effective across the majority of cases, just for high vision loss cases. That's what the data here supports as an argument. Okay, so Eylea is the most effective for high vision loss patients as there is no error bar overlap for 2 years. But in medium loss vision patients, there is overlap with Lucentis after 84 weeks, so no difference. This suggests Eylea is not best for the majority of cases, just high or only high vision loss patients. Okay, so we're on to our final part of this question, and it's another maths question. So the average dose given in an injection is 2.5 milligrams. In the UK, a 100 milligram vial of Avastin costs £242. Reportedly, Lucentis has a higher affinity with the VEGF protein found in the eye, but it costs £742 per dose. A dose of the newest drug, Eylea, costs £816 as it is an artificially made protein. Calculate the percentage saving in costs when treating 200 patients with the Avastin instead of the Eylea. So we need to work out how much each dose costs in Avastin. We've been told the dose cost for the other two drugs. So then we need to work out so times it by 200 to figure out, like when we talk about patients, we need to know what 200 doses would then cost. And then we need to work out the difference as a percentage. So Avastin, 100 milligrams divided by 2.5 milligrams tells us how many doses we'd get out of that bottle or vial, which is 40 doses, which is £242. So £242 divided by the 40 doses gives us £6.05 per dose, which is clearly much more sensible than £816 a dose, which is what the Eylea is. Because it's an artificially made protein. So although we've just said about Eylea being really effective for those patients with high vision loss, it's clearly very, very expensive compared to Avastin. So for 200 doses, then we need to do 200 times £6.05 and 200 times 816 to get what it would be to cost 200 patients, to treat 200 patients with this. So £1,210 for Avastin and £163,000 200 with Eylea. So now we just have to work out £1,210 as a percentage. The difference between those two as a percentage. So we need to do 163,200 minus 1,210 to get the difference between them, divided by 1,210, because we're obviously talking about how much more it would cost if we had the larger number, so that's why we divide by the smaller number, times by 100 gives us 13,388% saving by using the Avastin. So then this would be obviously something that the NHS or whatever would have to try and work out, whether that saving was worth it based on how much significant difference is there between the treatment effects of Avastin versus the Eylea. Is it worth spending money on the Eylea for really seriously high vision loss patients? Or is it worth just giving everyone and making Avastin available to everybody, and it would improve their vision, just maybe not as much? Okay, so the mark scheme for question 5 then. As we said, there's sort of the idea in the first part of the question, in part A, that we were talking about why visual acuity is worsened if there is bleeding in the fovea. And we have to say that there's a higher proportion, or there's a higher density, or that there's lots more cones in the fovea. That's the first part. And then the second mark talking about why that is linked to visual acuity. Well, because there's separate neurons to the brain for each cone cell, or there's separate bipolar neurons, so there's no summation of impulses, or impulses sent from each cone are kept separate. There's a lot of options there, but as long as you've got that as your second part of your mark, then you can have 2 marks. The maths question, you get 1 mark for calculating the 2 areas correctly. You get 1 mark then for calculating the answer. So obviously if you got 2.43 times 10 to the 5, then you can have 2 full marks. If you got the areas correct, you can have 1 mark, even if you messed up the dividing for some reason, you can get 1 mark for that. Parasympathetic, as we said, because obviously we're relaxing the muscles to get the eye to dilate. Then preventing an influx of calcium ions is what we said. So this is the first mark, because obviously we're saying what happens if depolarization doesn't happen. So what is the first thing that depolarization triggers? It's the influx of calcium ions. So stopping that coming in. If you didn't say influx, but you said like that they were coming into the sarcoma, for example, you could have a mark there, but really that sort of phrase, an influx of calcium ions, we should know. Then we say what that does then. If we don't have any calcium ions, what will that stop? So it stops the movement of tropomyosin, or it prevents unblocking or exposing of binding sites on actin, and the myosin heads therefore cannot bind to the actin. That part there is all in brackets though, so really the main thing is stopping the movement of tropomyosin or preventing the unblocking of the exposing of the binding sites on actin. Didn't have to say that last part about the myosin heads to get the second mark. Then part D, as we said, this is kind of a antibodies shape fitting together question. You need to say it's got a specific tertiary structure, or a binding site that's specific, or its variable region. Then you need to say its complementary shape to the VEGF protein. Or if you didn't say that, you would say it would bind to the VEGF protein. You can't say same shape, because remember, like puzzle pieces, if you've got 2 puzzle pieces that are the same shape, they won't necessarily fit together. You need one piece to be complementary shape to the other so that they fit together. And then, as I said, the last mark was saying, well, what does it do? How does it stop that VEGF from working? It prevents it from binding to receptors, or it prevents it from activating blood vessel growth. So if you just said it stops VEGF doing its job, as in it stops VEGF activating blood vessel growth, then you can have the mark. You didn't have to talk about it binding to a receptor, because that was sort of what I was thinking about, why the antibodies would help and stop it. But you can just say it stops it activating blood vessel growth. The question about why you can't be a pregnant woman and take part in this trial is because it could affect the growth and development of the fetus, or it could affect the growth and development of a named organ, so the lung, the blood vessels, the kidney, the brain, in the fetus. The main part is that you're focusing on the fact that it's affecting the fetus. It's not affecting the mother at all, because that's why it's asking about pregnant women specifically. So it was that sort of key part that you needed to get a mark there. As we said, the three arguments that we're going to use to talk about the evaluation of that statement. So the error bars do not overlap at any point for ILEA, for high vision loss group, over the 2 years. Less difference in effect, or there are some error bars overlapping after 84 weeks with ILEA and the Lucentis, and therefore the conclusion is not better to be used in the majority, as it's more effective for high vision loss only, not most effective across both groups. Something like that would have been fine for Mark 3. And then, as we said, you need to work out the doses. That gets you 1 mark. So how much per dose? £6.05 per dose for Avastin, and then working out the percentage gets you the 2nd mark. Okay, so that is the end of our exam style question part of the paper. And now I'm going to move on to looking at the essay questions. These questions in this paper, like I said, are big and synoptic like this, and they jump around all over the place and require you to use all your different knowledge from across the spec. I would say if you are someone that writes slowly and finds these application style questions hard, you could spend more time planning your essay at the start of the paper. So go to the paper 3, turn to the back, look at the 2 essay questions, plan or briefly have a think about both, maybe plan both or plan one briefly and really decide what you think you're going to be able to write about better. Then pause, go back to the start of the paper, do the exam questions. Like I said, if you are going to spend ages on the maths questions, skip them for now if you need to. Then come back to your essay questions and you can have a quick brief look over your plans. There may be things in the questions that have popped up that you've answered to the papers that have triggered your brain and made you think about some content that you could add to your essays. So that's always nice and helpful. And by that time, you've also had time to think about and really decide which essay you're going to go for. Then you can spend time, sort of 10 minutes doing a really proper plan and about 30 minutes doing the actual essay. 25 to 30 minutes writing the essay. So it means that you can plan ahead, spend less time choosing, panicking and planning over the essay at the end of the exam or towards the end of the exam, but you still make sure you've sandwiched in time in the middle to answer a lot of these questions and get marks as well. Okay, so we're going to look at the 2 essay questions in a minute. I'm going to plan both and then write both and go through my essays that I write. The main thing I want to do before that is to focus on this assessment guidance. So this is really helpful. It's from the mark scheme. If we're aiming for this high bracket, so the section where we're going to try and get over 20 marks, then we need to have an essay that flows and is interlinked with points that all contribute to the theme of the question. There need to be clear links between at least 4 topics, which is a minimum. The biology needs to be detailed and comprehensive A-level content beyond GCSE level. There needs to be no significant errors in that biological content and no irrelevant material that do not link together to the question. And if you want to get 24 or 25 marks, then you need to have evidence of wider reading by adding information that is relevant to the question, but not required by the specification. So not one of the bullet points or incorporated into the specification from AQA. That means it can be from another exam board and still be A-level content. That's fine. It must be at least 3-4 lines and not just single sentence fact. So this is all the things I've been thinking about when I'm planning and writing my essay. If I'm going to choose which essay I'm going to write, can I confidently cover at least 4 topics at a minimum? And for all of those topics, can I give the same level of detail, A-level content that I know without making any significant errors? And does everything I'm going to say or that I know about that topic, can I relate it to the question? Once you've done that, you can start, and you're only going to be able to do that by reading the question and listing bullet pointing some topics that you think you're going to include, okay? And then once you've done that, you can decide, okay, could I talk about all of those 4 to 5 things in lots of detail? Can I bring any other points that link into it, that link to the question? And is there anything beyond the spec that I know that I can link in here? Okay, so that's where we're going with this. Obviously, it's still quite sort of easy, I would say, to get into that bracket where it's about 16 to 20 marks. I would say that's probably the average mark base that people were trying to aim for rather than trying to get into this over 20 mark. I would say you've got some points that are detailed, but there may be some which are less well developed. So this is where you can end up spreading yourself a bit too thin and either mentioning loads of things and not going into much detail and not developing that really high level A-level content in there and just superficially explaining them and not actually going into much detail. And you are allowed like one significant error, so something fundamental that's wrong with your biological knowledge, or, and there's a big point there, or one irrelevant topic which detracts from the quality of the answer. So you can either have one significant error where you really, or you can have one topic where you talk about a topic for a while but it's not relevant. And that will still allow you to get into these brackets, but it may get you a kind of lower to medium answer. And that's what I would say. Try not to spread yourself too thin with too many topics in superficial detail, and try not to make sure that all of your topics that you are going to pick are going to relate to the question. Okay, so going on to section B, and we've obviously got to choose one answer in this section. I'm obviously going to do both for you, depending on which one you've done, so you can see how I've answered this question, both of these essay questions. Question 6 is the importance of organisms being able to respond to their internal and external environment. So we have a two-part question here. Response to the internal environment is homeostasis. Response to their external environment is things like behavioural responses, taxis, nesis, tropisms, and large things like population responses to the environment. So how does the environment impact population dynamics and things like that? This is something that also comes up. So the idea of the importance of is really common at the start of these essay questions now. And it's because it's the thing that tricks people out in that they think this essay is a way of showing off and just writing loads of content, showing off what they know and being able to write, just free write about topics and showing content knowledge. It's not. It's about taking your content knowledge and applying it to the idea of why is it important to the organism. So in this case, it's going to be things like survival, growth, maintaining your internal environment to make sure your enzymes don't denature, all of that. And as I'm going to say in both of these examples, plan, make sure you plan, but I would spend time going to the essay questions at the start of an exam, sit down with the paper 3 exam, go to the back, look at these essay questions, write brief bullet point lists of general big topics you would probably include for both, leave it, go back to the start of the paper, do the questions. When you've got about 35 to 40 minutes left of the exam time, hopefully you might have even more if you finish the questions quicker than that. Then come back and then look at your 2 bullet point plans. You may want to add things that you've discovered from the questions, which is great if they've helped you think of extra content. Then really decide, if you can, which one you're going to write more about, which one you're going to write more about in A-level level detail. Can you write about all of the topics equally in terms of detail? And can you think of importance links for every bit of the topic? Because if you can't, then there's no point in writing about it. So for example, I obviously am going to pick control of blood glucose as one of my topics here because that is a homeostasis topic. And the importance for that is, I already know it's about maintaining the water potential of blood, which is obviously really important, and maintaining enough glucose available to cells for energy, for respiration, to provide enough energy. I've put a link to adrenaline there because I've already thought about the fact that I could link to fight or flight response here and adrenaline as well, and the use of glucose, which is an external response sort of. It could be a fight or flight response. Thermoregulation, again, another homeostasis one, not necessarily one that is in much detail in this specification, so could be including some extra information from beyond the spec here, potentially an opportunity, but again, regulating the temperature is important because of enzymes. You can also link to external responses here, to their external environment, by linking to behavioural adaptations of things like ectotherms. So cold-blooded organisms, how do they respond, and plants as well. So that leads me nicely into my third paragraph, which is basically going to be a plant responses paragraph, which we can do a bit of both. So we can talk about responding to light and tropisms, and we can talk about responding to like temperature and light intensity as well, in terms of maintaining water content and transpiration. So that is a whole nice, lovely, I'm hoping going to be linked paragraph where I can link this idea of temperature or light and tropisms growing towards the light. Why is that important? Photosynthesis. Okay, and doing photosynthesis we need water, and also we need carbon dioxide, so we need to do gas exchange. But the balancing of the temperature response and the gas exchange, opening, closing stomata, and losing water is really important. And then we can link about the idea of obviously transpiration being affected by external factors and how they respond to it by closing stomata. I think that's a really nice sort of plant paragraph there where we can get a lot of topics in that should all be related to the question and will be nicely linked together as well. And obviously our importances, as we said, photosynthesis, water potential, so water loss and maintaining the sort of turgidity and support in water cells. Then my kind of fourth big paragraph is going to be about sort of one of those kind of broader topics in terms of population changes. So responsive populations to changes in their environment, i.e. natural selection, and how that ends up changing allele frequencies. And the importance of this one's quite nice because it's quite easy to talk about the fact that obviously this allows over time organisms that are better adapted to survive and reproduce and therefore pass on their DNA, which is the main goal of all organisms, is to be able to reproduce and pass on their DNA and have that kind of level of fitness that we talk about with passing on their genes. So there's kind of four broad topics. So I know at minimum I've covered four things. I already know I'm including at least two spec points if I talk about transpiration and tropisms, and maybe gas exchanges, 3 in the plant paragraph. So I'm going to have to be careful about how much detail I include. When I'm writing it, I might change and not include all 3, depending on how kind of quickly I think I can write this. Being aware with thermoregulation that I have to make sure it's like A-level detail and not GCSE level detail. Thinking about the importance of enzymes, I can link a little bit in there to enzymes denaturing and what I know about that. And then the last thing to think about is obviously we want to try and get into that really, really top end bracket of the mark scheme. So I'm going to need to include some beyond the spec information. So one of my main things I'm thinking is statoliths, because statoliths and gravitational responses are kind of important in plant responses to light. So I could mention that, and those are not on the AQA specification. The other thing I could think of is C4 photosynthesis. So if I'm talking about temperature and light responses, plant responses to temperature, and thinking about closing stomata, I was thinking about C4 photosynthesis adaptations. So how do we keep oxygen away from the visco, and how do plants in hot desert environments have adaptations to allow them to respond better to the environment? So they're both going to be potentially included in paragraph 3 or 4, potentially. I'm not sure yet. But I've got an importance for each one. I've got at least 7 topics, if I think about it here, covered by all of these points. So I'm definitely going to be covering more than 4. It's now about whether I narrow it down and make sure I can give enough detail about all of these topics to warrant putting them in. I need to pick one beyond the spec topic, really. I don't need more than one. And I need to think about which ones of these I'm going to really focus in on to get detail. So I'm going to write the essay, we'll go through my written essay now, and we'll kind of talk about making sure I flag up to you where and how I've linked all of these topics to the question and make that really clear to the examiners. An organism's ability to respond to their internal and external environment is vital for survival. Being able to respond to internal changes away from the optimum through negative feedback allows maintenance of the internal conditions that are required for many metabolic processes to function. Responding to external environments allows organisms to move and make sure they are in an environment that will provide them with all the resources they need, or to prevent them from coming to harm. Glucose is constantly needed in cells to produce ATP and provide energy, and it also contributes to the water potential of blood and body fluids outside of cells. Blood plasma is 90 to 92% water with dissolved solutes, including glucose. Blood glucose concentration must therefore be controlled to maintain the water potential of blood. Blood glucose concentration should stay between 4 to 8 millimoles per dm cubed of blood. If the concentration is too low, cells may not have enough glucose for respiration and may not function normally. Brain cells are especially sensitive to this. If blood glucose levels are too high, it can disrupt the water potential of blood and cause disruption as water will start to move in and out of cells due to osmosis, and this could cause cells to swell and lyse. In patients with diabetes, a very high or low concentration of glucose can even cause death. A negative feedback system works to maintain the concentration of blood glucose within the optimum range. Receptors in cells in the islets of Langerhans in the pancreas detect increases in blood glucose, for example after eating a meal, or decreases in blood glucose, for example during exercise, and respond by releasing hormones. Receptors in beta cells detect the increase in blood glucose concentration as glucose moves into beta cells by facilitated diffusion. This causes insulin-containing vesicles to move towards the cell surface membrane where they release insulin into the capillaries by exocytosis. Insulin binds to specific receptors on the membranes of target cells, including muscle, liver, and fat storage cells. Insulin binding stimulates the cells to add more glucose transporter proteins to their cell surface membrane, which increases the permeability of the cells to glucose. This increases the rate of facilitated diffusion of glucose into these cells, which lowers blood glucose concentration. Insulin binding to receptors on liver cells also causes the increase of uptake of glucose in the liver by stimulating glycogenesis, which converts glucose to glycogen. This helps to lower the glucose concentration in the liver cell, which maintains the glucose concentration gradient between the capillaries and the liver cells. Okay, so main things I want us to take from this paragraph. I did a very brief introduction. You do not need a really long introduction talking about what you're going to say. There is no point, there is no time. It's not worth repeating yourself if you're going to just say what you're going to say later on. And it's not worth talking about things that are not going to get you marks because they're not relevant to the question. The only thing I would say here is just make it a brief kind of couple of sentences on the importance of responses as a whole, I would say, which is what I try to do here. Just to kind of get yourself, it's sort of almost a signpost to the things you are going to talk about in terms of the importance part. And that's obviously what we're trying to make sure we do. I actually spent a lot of the time at this whole section, at the start of this paragraph, talking about the importance of regulating blood glucose. It doesn't really matter where you put the importance that you're going to talk about, just make sure you obviously include it. And sometimes if you put it at the start, it means you don't forget it, talking about it at the end, and then you run on to something else. So this is why I put it here. It also just makes sure that I've covered it, and then it feeds nicely into kind of like, this is really important, this is why, what can happen if it gets too high, what can happen if it gets too low. This is really focusing on the importance. And then I've got a nice chunk of correct A-level content. So this is not the type of blood glucose concentration paragraph you'd expect from a GCSE student. So it's got a lot of the A-level specific content that we have learned. Also, you'll note that I've talked about receptors. You need to be mentioning receptors and how these changes are detected all throughout this essay if you can, because that is obviously part of the response, okay? So yeah, definitely kind of make sure we've got the importance covered, make sure we've talked about detecting the changes, what the change is, and then how they respond to it, and we've covered why that response is then important in the body. Okay, so this paragraph is kind of an extension of the first paragraph, although it is a different spec point and counts as a different topic. I wanted to make sure I included this section about adrenaline because it kind of links what we've just been talking about with blood glucose, but also kind of an external environmental response because it is a response to danger and fight or flight. And also it's a way of talking about the opposite of what we just said in terms of increasing decreasing blood glucose, we can now talk about increasing blood glucose, but without just talking again about glucagon, because then obviously that's just talking way more about the same thing in the same topic, which I think is a bit unnecessary here, which is why I picked one, and then this one allows us to do kind of both, but whilst talking about a secondary topic and talking about more A-level kind of detail level content, but also talking about kind of an external and internal response at the same time, so it covers both points of the question. When blood glucose levels are low, the hormone glucagon is used to increase blood glucose, but the hormone adrenaline can also act to increase blood glucose levels as part of the fight or flight response to danger detected by audio or visual receptors. Adrenaline is released from adrenal glands into capillaries and binds to receptors on liver cells. This causes the enzyme adenyl cyclase to change shape and become activated. Active adenyl cyclase catalyzes the conversion of ATP to the second messenger cyclic AMP, or CAMP, which activates a protein kinase enzyme. This triggers a cascade which catalyzes the breakdown of glycogen to glucose, glycogenolysis, and inhibits glycogenesis in the liver and muscle cells, so that more glucose is available to muscle cells for respiration, so that the body will have enough energy the body will have the energy source it needs to run or fight the danger. This is a vital response which helps keep organisms safe from danger. Okay, so my importance there is at the bottom, and I've linked it directly to this idea of needing to have that extra glucose for respiration in muscles so that the body can have the energy source needed to contract the muscles, to move the body, to run or fight the danger, and obviously that keeps the organism safe or increases the chance of survival. And so that's kind of the importance of this response to danger. I mentioned the audio and visual receptors. So obviously detecting danger is normally something you see or you hear, especially if it's like you're a prey animal, they're going to see or hear sounds that are going to trigger this response and increase their adrenaline. You could have talked about other things that adrenaline causes, changes in the body, like heart rate, breathing rate as well. I've gone into a bit more detail with this, but this is sort of an extra topic that we're including as on top of the kind of main four. So I didn't want to go into too much detail and not have enough time to make sure I covered all the topics. Okay, so moving on to, this is now our third paragraph, but our second kind of big response topic. Another system that helps to regulate the internal environment in animals is the nervous system. One example of the action of negative feedback involving the nervous system is in thermoregulation. Regulating body temperature is vital because all the metabolic reactions in animals involve enzymes, and these are temperature sensitive. If the internal temperature is too low, then there will not be enough kinetic energy and the rates of reaction are slow. But if the temperature becomes higher than the optimum, then bonds holding the tertiary structure of the enzyme in place will start to vibrate and eventually break, denaturing the enzyme and changing the shape of the active site so that it can no longer form ES complexes, and enzyme-controlled reactions cannot continue. In humans, the hypothalamus in the brain contains the thermoregulatory center. It contains receptors sensitive to the temperature of the blood. It also receives nervous impulses from thermoreceptors in the skin, and then sends impulses along motor neurons to various effectors. If the temperature of the blood is detected to be too high, sweat glands in the skin are stimulated to produce more sweat, which evaporates using heat energy from the skin and transferring it to the environment. Vasodilation occurs to allow more blood flow to the skin capillaries, which allows more heat energy to radiate from the skin. Electrical impulses are also sent along nerves to the hair erector muscles in the skin, which relax, causing pylorelaxation. The detection of high skin temperatures can also trigger behavioural responses to high external temperatures, such as cooling off with water, seeking shade, or reducing activity to reduce the heat generated through respiration. Some organisms can also increase their surface area to increase heat loss through conduction. This is especially true of ectotherms, who are not able to internally regulate their temperature and must rely on absorbing heat energy from or radiating heat energy to their environment. So in this paragraph, I've managed to talk about the nervous system, which obviously is different from the endocrine system. So we're covering different spec points here. I've talked about thermoregulation, some of which is explicitly stated in the spec, but some of it is not necessarily. I've talked about electrical impulses, the idea of receptors, motor neurons, effectors. We've talked about how they actually change their temperature. And then we've also linked that to external temperature as well as internal temperature regulation by talking about behavioural responses as well. So, and we've talked about like ectotherms and who can't, regulate the temperature. All of that is great stuff. All of that is linked to the nervous system. It's got internal and external kind of responses to stimuli in there. And it's a contrast and is covering more spec points, but it is all linked and related to this idea of responding to the environment. And we spent a lot of the time at the beginning talking about enzymes, which included A-level content about enzymes. So we didn't just say they denatured. So we linked our importance to some A-level content about enzymes as well, which is always really good. And we spent a bit of time talking about that before we went into the actual how we change the temperature in response. So we're on to our kind of third big point, fourth paragraph here. This is obviously taking that sort of paragraph from before, which was talking about the nervous system. We're running it into this one saying plants don't have a nervous system, so this is how we're going to respond. This is also where I'm going to include my extra content. So I'm going to highlight that in a different colour because this is my beyond the spec content that I'm including here. If you took that out, this obviously essay would still be a decent essay and would get you into that high level bracket. But as we said, adding in that extra beyond the spec content is needed to get you into that 24-25 mark. So it's only sort of short, but it needs to be more than a sentence, remember? So there's a couple of little bits in here that are directly linked to the expression of genes and how that the environment can have an impact on gene expression, which could be a whole other paragraph talking about epigenetics if you wanted to. That is obviously a way that the environment leads to responses in organisms. So I've kind of linked two sort of extra content plus another sort of spec point in here by including that together. So this is kind of a plant-based paragraph, but I did that and added that extra content in and talked about tropisms rather than focusing on trying to do tropisms and transpiration and gas exchange, which I think was too much from my plan. I've trimmed it down to just tropisms, but talked about two types of tropisms and linked into that extra content as well as another spec point. Plants do not have a nervous system, so their response to stimuli are controlled by growth factors, which are hormone-like chemicals that speed up or decrease plant growth. These growth factors are produced in meristems in the tips of roots and shoots where cell division is happening. IAA is an auxin that is produced in shoots and roots and is transported to different parts of the plant to control tropisms, which are responses to directional environmental stimuli. These tropisms are important as they allow plants to increase their absorption of essential resources required for photosynthesis, including light, carbon dioxide, and water. Shoots are positively phototropic and grow towards the light. They can sense the direction of light using photosynthetic pigments and grow towards it to maximize light absorption for photosynthesis. IAA moves to the more shaded part of shoots. This increased concentration of IAA on the shaded side promotes cell growth, causing those cells to elongate so the shoot bends towards the light. This is an example of how gene expression can be controlled in response to the environment, as it is thought that light intensity affects the expression of genes that code for PIN3 protein pumps, which transport auxin across the stem or roots. The distribution of protein pumps due to light intensity will result in the auxin gradient that leads to change in the growth of the shoot. Negative phototropism and positive gravitropism are important responses to help ensure plant roots grow deep into the ground. This ensures that they can absorb enough water and mineral ions required for plant growth, but also so that they are able to compete with other plants for if these resources become limiting. Deep roots also act as an anchor to protect plants from the actions of strong winds to make sure they remain upright to ensure they can absorb enough light for photosynthesis. Plant cells use organelles called statoliths, which are starch-filled grains that detect the direction of gravity. They accumulate on the lower side of plant cells due to gravity. This results in high concentration of IAA at the lower side of the root, which inhibits cell elongation. The gradient produced causes the cells on the upper side to divide, and it elongates faster than the lower side, causing the root to bend downwards. Okay, so you can see here that I've got kind of those areas in green where I talked about things that are beyond the spec. So statoliths and the pin-3 protein pumps and the epigenetic control of those are not obviously part of the specification, so I've added those in extra. It's more than a couple of sentences or more than one sentence, and it's directly linked to what's happening and what I'm explaining and related to the question. Obviously, I've also mentioned the importance of both gravitropism and positive phototropism, and also the general importance of tropisms in plants. And I sort of talked about the fact that they can detect light using the photoreceptors. There obviously aren't really any receptors with gravity, but that's why I talked about the statoliths. And then we're talking about the fact that IAA is a kind of hormone-like growth factor. It's produced, it gets moved around, and then the response that it causes in cells. There could have been even more detail here if you talked about IAA. You can actually talk about the fact that it helps to loosen the, sort of increases the stretchiness, the kind of elasticity of the cell walls, and that helps with elongation and things like that. You could have gone into a bit more detail here as well, but this is enough. It's quite a big paragraph. It's covering tropisms. It's talking about plant responses. We've got the importance in there. We've got our beyond the spec content in there. So it's answering the question. Okay, on to our final paragraph, which is kind of one of the more broader answers to this question, thinking about how the environment causes a response or a change in the allele frequencies of a population through natural selection. So variation in organisms can be caused by a combination of both genetic and environmental causes. Environmental causes of variation can often be described as selection pressures. Changes to an organism's environment can lead to a change in allele frequency in the population through natural selection. Organisms with variations that make them more likely to survive are said to have a selective advantage. They're more likely to reproduce and pass on those alleles to the next generation. So these favourable alleles increase in frequency in the gene pool. Evolution is the change in the frequency of a gene in the population. If selection pressures are different for different populations, then evolution can occur and new species can emerge once the populations can no longer interbreed. These populations become adapted and specialised to those environmental conditions. This response to environmental change is important as it allows populations to survive and continue to pass on their DNA to the next generation. An example is directional selection in peppered moths. Before the Industrial Revolution in the UK, light-coloured moths were better camouflaged against clean silver birch trees, which allowed them to avoid predation by birds, so the allele for light colour was more frequent in the population. As the Industrial Revolution produced more city trees in England, dark-coloured moths became the better camouflaged phenotype in the new city environment. Due to natural selection, the peppered moth population responded to the change in the environment by shifting the allele frequency so that the dark phenotypes were more common. Okay, so that is it. We've talked about, so briefly there, mentioned the importance. So talking about the fact that obviously they're passing on their DNA, it allows them to survive. This is the thing, it allows them to survive and cope with change, which was also mentioned kind of a bit later on as well. So it's one of those kind of a bit more abstract answers to this question, because there isn't really a direct receptor that's detecting a change and then there's a direct response. This is kind of a long-term response to changes in the environment by populations over time. So this may feel slightly different and awkward, and it may not have been your first choice. It's just me doing this paragraph as kind of show you an idea of what it means to do something quite big and broad with some of these kind of what seem like very specific questions. So hopefully that was helpful, and you kind of seen what that can be included and kind of the level of detail and A-level detail, because again, this could have been a GCSE answer, but including things like talking about the gene pool, talking about the change in allele frequency, talking about selection pressures, speciation, this made this clearly an A-level answer to this paragraph. So hopefully this kind of level of detail has been really clear about how much detail you need to go into for all of your points, and how we must keep that detail at A-level detail and not just superficially mention a load of things and not really go into detail and not really link it to the question. This paragraph, I would say, it's harder to link to the importance, but obviously we did have that kind of extra paragraph with adrenaline. So I would say I'll be confident that across the 5 sort of big topics we mentioned, there's enough A-level detail there, enough linking to the question to get us into that top band of marks. And that's what I think is really important, and why sometimes it's actually more important to think about sort of bringing in other topics to paragraphs and making sure there's at least 5 sort of spec points covered with what you're writing. And then you've got that time and space to go into that extra level of detail. So we're going to have a look at the sort of, not the mark scheme, but the list of things that they said you could include other than what we've talked about, and also some kind of do's and don'ts for what examiners have said was weak in terms of performance on this type of essay question in the past. Okay, so as I said, the mark scheme for these questions, we've looked at the mark scheme already, and that's what you would use to have a go at marking them, but they also give you these lists of kind of topics that you could include, not exclusive, so you don't have to include only these, and if you did anything else, it wouldn't be correct. These are just kind of like the main topics that they would sort of be expecting you to use. So we obviously talked about a bit about behaviour, we talked about adaptation and selection, we talked about changes in populations and selection pressures, we talked about response to stimuli, plants and tropisms, we talked about taxis and kinesis, and we talked about homeostasis response to changes in internal environments. So as I said, we've got quite a lot of things covered there, because it's also hormones and responses. There's loads of that we've covered. Gene expression as part of response, a little bit, probably not enough to count as a whole topic, but we've been broad with our response. And there's some other things that we didn't even mention, so control of heart rate is a good one. We've also got the immune response, which is a nice one, because obviously that's responding to pathogens and how that helps keep the body safe. Transpiration, as we said, it wasn't our plan to do that in gas exchange, but we didn't end up doing it. But that would have been a nice one, because it's obviously a response to environmental factors. So there's loads of things you can talk about. So, and there's others as well that aren't on this list. What I would say, and what I have done, is had a look at the examiner reports for essay titles similar to this, and looked at what they've said students did poorly on, so what a lot of students failed to do, and what we should avoid doing. So this is what I've got on my list here. So these are things that if you are trying to mark your essay, obviously remember we need to make sure we're not doing irrelevant information, we're making sure we're not missing out information or not answering the question or getting anything wrong or incorrect or not going into enough level detail. And that's basically a summary of what all of these points are. So if you're finding that you're doing any of these things, or you reread your essay back to yourself and you feel that actually these are the things you're doing, this is the way to try and improve and avoid doing this in the real exam. So not focusing on the importance of the responses, typical. So for any importance question, that is going to be the main one that students fail to do. Not including broader examples like populations and the adaptation and selection like we did. Again, it's a bit left field and it's a bit out of the box, but it helps you kind of really make your essay kind of broad and include kind of topics that are relevant, but you just wouldn't necessarily think of them straight away. But as long as you can turn them around and make them link to the question, then they're good to include. Not focusing on receptors, so not mentioning receptors in any of this, and so how are they actually able to respond to the environmental changes? How are they sensing the changes in the first place in order to respond to them? Not linking transpiration to a response in environmental factors, or wandering into discussing irrelevant detail like roots and absorbing water. So this would be like picking transpiration and then really not making it a response to environmental factors, because it's not an internal response, it's an external response. Lacking factual A-level detail of processes, so only superficial descriptions or GCSE level detail of homeostatic processes. So this is obviously everything that could be included in GCSE, like the blood glucose, like the kidney if you did that, even tropisms as well. All of that is covered in GCSE detail in some point. So not going into A-level detail and just skimming across it and talking about loads of different homeostatic processes, but without that really critical level of correct A-level detail. Lacking detail on thermoregulation with errors and lack of receptor detail, so missing out the idea of receptors and how you detect temperature in the first place. can make that less sort of important to the question. Not giving enough detail on thermoregulation, again, this could be a GCSE problem as well. It's not really covered properly in the spec, so it's one of those ones where you have to know that detail properly. Superficial GCSE level detail of taxes, kinesis and tropisms, incorrect examples and little detail on importance. So mixing up taxes and kinesis is a common one, not giving importance. So we also did tropisms and didn't really talk about taxes and kinesis much at all, other than the behavioural response to temperature. But if you are going to go into detail of taxes and kinesis, it's really important to make sure you've linked that idea of importance. So thinking about wood lice moving in a certain direction because they don't want to lose water loss, for example. Failure to talk about true reflex responses. So if you're going to do reflexes, neurons and synapses, and talk about reflex responses, and the importance is obviously they keep organisms safe from danger, then you are going to actually have to talk about a true reflex response and not just, oh, they can see danger and run away. That's not a reflex response. You need to think about things like pupil dilation, etc. Poor language use, for example, organisms needing to or having to evolve. So you can see how that could slip in if you are really trying to think of an importance for that last question that we talked about. So not thinking about it as a whole population response to change over time and change to the environment over time, but thinking about, oh, well, organisms need to adapt or they have to evolve in order to survive. The organisms don't know they're doing that, and it's not something that's happening at an organism level. It's happening at a population or like a whole species level. Okay, hopefully that was helpful. We're going to do exactly the same thing with the other essay now. So even if you didn't do that one, I would suggest, especially with these papers, because you've got this option here, do both essays. Don't do it in your timed practice of this paper, but pick whichever essay you're going to do and do it, and then mark it. And then on another time, set yourself 40 minutes to plan and do the other essay and still have a go at marking it using, or go through it with me afterwards as well, using this method. Okay, so the other essay option we have, which personally is my favourite, and I think if I had been doing this essay, this is the one I would have chosen. So write an essay on the importance of energy transfer within and between organisms. So this is really important. It's got two parts. So energy transfers is the focus. So energy being transferred from one form to another. Light energy to chemical energy, for example. Chemical energy to kinetic energy. And that's got to happen within, so inside organisms, and between organisms, so from one organism to another. So for example, feeding, where one organism is going to eat another organism in order to get chemical energy, which they then use to help them produce their own energy through respiration. The importance is really, really key. And this is why a lot of these essay questions are using this word now at the start, because you need the why. If you're talking about, it's not just a case of just tell me everything you know about energy transfers and that topic. You need to say why these energy transfers are important with kind of scientific knowledge. So my plan, I'm going to try and focus on the paragraphs I'm going to write. And I think there's kind of three main areas, but they will probably end up as larger paragraphs or potentially more paragraphs. So I'm thinking about kind of starting, going through the food chain almost. So start with producers, because that's where light energy is converted into chemical energy. So start with photosynthesis. It's really easy to think about energy transfers in there because we've got electron transfer chain, we've got photolysis, so light energy is being used to split water, the electrons passing along the electron transport chain, the energy is being used to create ATP, so it's photophosphorylation. The ionizing of chlorophyll, so that energy from light is used to transfer the light energy to excite the electrons, get them to a higher energy level so they can leave the chlorophyll. All of these are energy transfers. And if you make sure you talk about them like that as you write them, that will be answering the question. The importance of this, and this is the thing I will tell you, is absolutely necessary. When you are planning your paragraphs and planning your essay, everything you write must have an importance link next to it. Because if you can't think of importance, it shouldn't be in your essay. And if you can't think of an importance, or you don't write one down, you won't put it in the essay, and that means that section of your essay will be counted as irrelevant or not linked to the question. So we can't get into that top mark bracket. So obviously the importance of photosynthesis in general is it's how producers start all food chains. They are the source of chemical energy for every consumer. So if we didn't have plants doing that process, we wouldn't have chemically available energy in the form of glucose. So then I'm thinking about transferring from producers to consumers, because that covers my between organisms, and I get that in early in the essay, which means I'm definitely going to be able to have time to cover it. So food chains, transfer of energy through digestion of biomass, the wasting of energy, how energy is not transferred through those food chains and it's lost to the environment. We can talk about GPP and MPP in farming. This is all making this A-level standard, because remember, we could just end up talking about food chains like a GCSE level, and we don't want to do that. So GPP, NPP, or GP and NP, farming, the conservation of energy through farming practices to make sure we're increasing the energy transfer in food, energy losses through heat, respiration, not eating all of an organism, why energy is lost at each level of the food chain. Why is this important? Well, it's important for the food chain of humans. It's important to make sure that if we're doing these practices in farming, we're trying to get as much energy transfer into the food and that we're eating as possible. It's also kind of a link to other things like the carbon cycle, kind of energy moving through, and then energy that's wasted and obviously comes out in feces and stuff can end up going to decomposers and being recycled. But the other thing is obviously digestion and eating and consuming of organisms is really important, because that means we get the glucose in order to make ATP in respiration. And then that's a nice link then to our next paragraph, which will be respiration. So you can see here how I've decided to talk about my topics, and I could have bullet pointed just the topics like first is respiration, energy transfer, like all the topics from the energy transfer topic, and then sorted these out. So obviously this has taken a bit of thought on my part before I started writing this level of plan down. But that's what I'd suggest you do. You look at both of them on the paper, you write some sort of brief bullet points about the main big topics you think will be included, see how confident you feel about writing about those in detail, and then you make this plan where you get a bit more granular about what you're going to include, because you can't include everything. And then you can start thinking about how to order those paragraphs to help you make that nice flowing, fluid story that means that your essay is very clear and cohesive and one whole synoptic kind of essay, that all of it answers the question because you're going from one thing to another. So then I'm going to do converting chemical energy to ATP in respiration. So we're taking the chemical energy in bonds of glucose and we're converting it to the energy that's locked in the bonds of ATP. So we've got, again, similar process to the photosynthesis. We've got oxidative phosphorylation. We've got phosphorylating of glucose to make it more reactive. So the uses of ATP here as well. And energy losses here. So why is it not efficient? Why are the energy transfer in respiration not efficient? How can we end up we don't make the total amount of ATP we should. So the importance, obviously, is that you need ATP for energy requiring processes, which then takes us to more energy transfers, where everywhere where ATP is used, it releases energy and that energy is transferred to do something. So then you just need some examples of uses of ATP, because they're all energy conversions or energy transfers. So I think muscle contraction is a nice one, because you can talk about chemical energy going to kinetic energy to move and contract those muscles. So talking about the use of ATP to move the myosin heads and then detach the myosin heads and allow that shortening of the sarcomere, I think is a really nice one. Active transport, again, is another form of kinetics. So we're moving molecules from one place to another. So you've got the movement of glucose, active transport of glucose in the small intestine, which links all the way back then to digestion and food chains, which is nice. Or sodium potassium pump in nerves, maintaining concentration, resting potential. Again, really, really important. And there's nice importance links there. So muscle contraction is important for movement, which is important to be able to get prey and do the digestion and eating to get that chemical energy. Also things like the heart is a muscle, the heart contraction, that's important. It beats, it moves blood around the body, and that's important for delivering the oxygen needed for respiration and the glucose, which is, again, nice kind of cyclical story there. Active transport, sending impulses is really important, so being able to maintain resting potential so that then depolarization can occur, changing it, and therefore we can sense our environment, respond to the environment, or obviously absorbing glucose or mineral ions from plant roots and things like that, so getting the nutrients and minerals we need in order to then carry out these processes that require it, like respiration. So I'm just kind of quickly adding up how many kind of topics I've covered or will cover in this, because I've got to make sure I'm covering 4 separate topics, which I am with my 4 paragraphs, but even within that, I've got kind of the food chains is one, and then the whole GPP and MPP and kind of farming is another one, because it's a whole other spec point. Converting chemical energy to ATP and respiration is a spec point. Talk about muscle contraction, spec point. Active transport, another spec point. And then I need to think about where am I going to include my beyond the spec information. Now, this is not vital. If doing all of this and getting all of this level of detail for all of this stuff to get into this essay it's still challenging. So there's not, I would say, unless you are someone who's got all of this content down and is really excellent at writing these synoptic essays, I wouldn't spend too much time stressing about learning enough beyond the spec content that you can have at your fingertips to apply to any essay question. I don't think it's worth the hours and the extra time that would do if you don't know all of the content you're supposed to know well enough and in enough detail to get it. You won't get into that top part of the bracket of the marking system anyway. But this is me doing it so that you can see what an example would look like. So one of the only main things I can think of here for extra content was talking about photorespiration. So photorespiration is not in the AQA spec. So talking about how that actually the failure of Rubisco to be not exclusive to be have carbon dioxide as a substrate, and how in high temperatures when oxygen builds up in leaves it can actually bind with Rubisco, and we do photorespiration instead, which limits the energy transfer. So that wastes the light energy that was absorbed, because products of the Calvin cycle are used to then regenerate and sort out photorespiration to actually make sure that we can still make glucose from it. So I thought that was nice because it actually links into this idea of energy transfers, and it talks about the lack of energy transfer efficiency in photosynthesis. So that's where I'm going to add my extra content in. Like I said, that would be where I'd go sort of as my last point of call if I knew I could confidently do it and if I knew it was related to the question. There is no point throwing extra facts into these essays if they are not directly related to the question. Okay, so I've talked a lot about this, but in reality, you think about that plan and how quickly that plan was. That plan was about 10 minutes. So I'm saying that plan would be about 10 minutes once you've chosen the question and having already thought a bit about what you're going to include. So this is why I'm saying start of the exam, go to the back, look at the 2 essay questions, brain dump your first thoughts or your first major topics, 4 topics that you think you'd include. So this one was obviously photosynthesis, food chains, respiration, ATP or ATP usage. That's my 4 things I would have written down. I could have written down some more and crossed them out, but that's what I would have written down at the start. I'd have written down some bullet points for the question 6, and then I would have just decided, gone and done the rest of the paper, come back with about 35 minutes left of the time left on the exam, unless I finished earlier than that, which is great, and then written out this plan, taking myself 10 minutes to write the plan, so I've then got 25 minutes to write this out in the detail that it needs to be written out in. Okay, so this is my first paragraph. I do have a very, very, very brief 2-sentence introduction. It's really not necessary, and do not waste time on an introduction, especially if you're going to end up talking about things that are not relevant or you'll be repeating yourself later on. Energy transfers are necessary in living organisms to maintain life, as all essential life processes and chemical reactions require energy in different forms. ATP is used as the biological energy currency, so energy transfers can be seen anywhere this molecule is produced or used. Energy flow through ecosystems begins with the capturing of light energy and the transfer into chemical energy in the form of glucose and other molecules, which make up the biomass of plants. This is achieved through the process of photosynthesis. The light energy is absorbed by photosynthetic pigment molecules arranged in photosystems found in the thylakoid membrane of chloroplasts. The energy from photons is used to excite electrons in a chlorophyll A molecule, which raises them to a higher energy state so that they can leave their electron shells. Light energy is also used to split water into protons, electrons, and oxygen. The electrons are used to replace those lost from chlorophyll A at photosystem 2. The excited electrons pass along a series of proteins called the electron transport chain through coupled redox reaction. The energy lost from these reactions is used to actively pump protons across the thylakoid membrane, creating an electrochemical gradient. This transfer of energy results in the movement of protons down their concentration gradient through the ATP synthase enzyme. This kinetic energy is transferred into the chemical bond formed between ADP and inorganic phosphate to produce a molecule of ATP. This process is known as photophosphorylation. Light energy is also absorbed at photosystem 1, where the electrons in another chlorophyll A molecule are excited and this time are used along with protons to reduce the coenzyme NADP. The reduced NADP and ATP made in these light-dependent reactions are then used in the Calvin cycle, where carbon dioxide is fixed by the enzyme Rubisco and combined with the molecule of RuBP. Through a series of reduction steps, including the transfer of energy from ATP, this molecule is converted into triose phosphate, which can then be used to construct organic molecules, including glucose. So main things I want you to get from this paragraph. I have really briefly mentioned the Calvin cycle, an overview of the Calvin cycle. I've not gone into detail as it's not relevant, and there wouldn't be many opportunities to talk about energy transfer there. I talked about most of my energy transfer in the light-dependent reactions that I've talked about. So don't waste time saying everything you know about photosynthesis here. It's not worth it. You need to make it linked to the question. You'll notice all the pink I've highlighted is everywhere where I've talked about transferring of energy. And I've been very clear to use those words, transfer of energy, everywhere I can. Talking about moving it to kinetic energy, talking about this transfer is used for movement, this transfer is used to do this, construct ATP. That's what this question is like getting at, and start strong with something like this where you can get loads of those really clear signposted into it. I needed to talk about the construction of organic molecules, including glucose, because that's going to link to respiration later on and also help me go into the next paragraph. All of that light-dependent reaction is the level of detail you need. It's A-level detail, it's correct, and it's linked to the question. This is what these essays are about. You describing steps, processes in really nice A-level correct detail that you show off what you've learned, but that it has to be relevant to the question. And that can't just be tagging a sentence on the start or the end saying, this is important because, and now I'm just going to tell you everything I know. I was very deliberate in my language here and how I wrote about this in order to make sure it was relevant to answering the question. So this is then my extra bit that I'm adding on to the end of this paragraph, which is my beyond the spec information about photorespiration that I spoke about in the plan. I've separated it because without it, this essay would still be high scoring, and you don't, as I said, need to really stress about including that extra content unless you are really super confident with all the content you need to know in order to be able to get high marks in the essay. This is just sort of icing on the cake, gets you that sort of 24, 25 marks out of 25. This form of photosynthesis, C3, is energy inefficient at high temperatures, as Rubisco combines to oxygen as well as carbon dioxide. In high temperature environments when stomata must close to prevent water loss, oxygen can build up in leaves and bind to RuBP instead of carbon dioxide. This is inefficient and wastes about 25% of the products of photosynthesis, and therefore the light energy absorbed to make them, as ATP and amino acids are needed to convert the products back to G3P for it to continue in the Calvin cycle. This process is known as photorespiration, as it uses oxygen and produces carbon dioxide. So that's it. It's more than one sentence. It's linked to the question because I talked about energy being wasted, light energy that's being used is being wasted to do this process. So it's linked to energy transfer and it's relevant and it's not on the AQA specification. Then I'm finishing my paragraph with an importance link to the question as like an overall sum up, ready to go into the next one. So despite its inefficiency at high temperatures, photosynthesis is a vital process to all life as producers are the source of chemical energy for all heterotrophic organisms. Okay, so this is a link back to the question, why is photosynthesis important? That is what that sentence says, and it's also going to link really nicely into my next paragraph about food chains. The efficiency of energy transfer from light to chemical energy, or biomass in plants, can be measured as gross primary productivity. Only about 1 to 5% of the light energy available from the sun is transferred to chemical energy in photosynthesis. A lot is lost through reflection of light, light not hitting photosynthetic pigments, and some light passing through plant tissues. Some GPP is also lost through respiration in plants. Farmers try to maximise the productivity and yield of crops to increase the energy transfer to humans. They do this by reducing competition with other plants, spraying herbicides, increasing plant growth by adding fertilisers to soils, and reducing yield losses to pests or pathogens using pesticides or biocontrol agents and fungicides. The transfer of chemical energy through food chains from producers to consumers is also inefficient, as only about 10% energy is transferred to each trophic level. This is measured as the net productivity. Some energy is lost as not all parts of plants or animals are eaten. For example, roots and bones and some biomass travels through the gut undigested and is lost in feces. Most energy lost is used for respiration, movement, or maintaining body temperature in consumers instead of being transferred to chemical bonds in new molecules to produce new biomass, which would then be available to the next trophic level. This limits the number of trophic levels in a food chain, as there is simply not enough energy available for organisms to survive past 5 levels, as the decrease in net productivity is too great. Livestock farmers can use the knowledge of energy losses to improve the efficiency of energy transfer to humans as the secondary consumers by restricting movement and keeping animals warm to reduce the amount of energy lost through respiration. Maximising the net productivity or net primary productivity available to the next trophic level means more light energy has been transferred and stored as chemical energy in bonds of organic molecules, which will be available to the next trophic level to be used in respiration, which is essential for producing the ATP needed for energy requiring reactions on a cellular level. So that last sentence is quite long. I think I should put more punctuation in there. Is an importance link. So it's importance of why making sure we get enough energy from our food, or making sure that there is enough energy in our food and maximising it through farming practices, is really important for humans. And then it links really nicely to respiration and ATP, which is what's going to happen in the next paragraph. This whole paragraph, you can see everywhere where I've talked about transfer of energy, this whole paragraph could be at a risk of being GCSE level content if it wasn't for me talking about net productivity and gross productivity, which is clearly A-level content, talking about minimising the respiratory losses, using kind of language like biomass, which isn't always used at GCSE as well, and just really kind of hammering home that idea of obviously chemical energy being locked up in bonds, chemical energy being available through glucose, and then talking about this idea of net productivity and what's actually passed on. So this is really important because it's the only paragraph here as well that links to this idea of between organisms. Unless you were going to potentially talk about digestion, you could go into a bit of detail there, but this was the easiest way I could think about to link it to energy transfer. Respiration itself also involves many energy transfers in the process of producing large amounts of ATP. In the cytoplasm, 2 molecules of ATP are used to phosphorylate glucose to make it more reactive and form fructose bisphosphate. As this molecule is now unstable, it splits into 2 molecules of triose phosphate, which are oxidized to pyruvate. The oxidation releases 4 molecules of ATP and reduces 2 molecules of NAD, which gives a net gain of 2 ATP for the process of glycolysis. The reduced NAD and the pyruvate enter the mitochondria, and the Krebs cycle is used to decarboxylase and oxidize compounds to produce more reduced NAD and FAD. The energy from the original glucose molecule is now stored in the bonds of these reduced coenzymes. The coenzymes travel to the electron transport chain in the cristae, where they are oxidized, and their lost electrons move along a chain of cytochromes through a series of redox reactions. This process is known as oxidative phosphorylation and is used to make ATP in the same way as photophosphorylation through chemiosmosis. It uses protons donated from the reduced coenzymes and requires oxygen to be the final electron acceptor in the electron transport chain. So briefly, I've written this note on the side. Do not waste time repeating exactly the same information that we talked about when we talked about photophosphorylation, because it's not worth your time and you've just repeated the same knowledge and content. So don't bother here. Make sure you're using your time to talk about something else related to energy transfer. So like photosynthesis, the process of respiration is also inefficient. In theory, 38 molecules of ATP can be produced from 1 molecule of glucose, because each reduced NAD should provide enough energy for 3 ATP, and each reduced FAD should provide enough energy for 2 ATP to be produced through ATP synthase. However, the net gain of ATP from 1 molecule of glucose is 32 ATP. This is because ATP is used throughout the process to transport ADP and pyruvate from the cytoplasm into the matrix of the mitochondria, and to transport ATP back from the mitochondria into the matrix to be used by the cell. Energy transfer is therefore incredibly important in respiration because the production of ATP is required to help the process of respiration to occur and produce large amounts of ATP required for many metabolic processes in the cell. Again, importance of respiration here, importance of energy transfer, why it's important to respiration, and then also why is respiration important? Oh, because it's producing ATP, which we need for metabolic processes, which is the link to the next paragraph. As I'm reading this, I could have made more of the use of ATP in respiration, talked more in detail about active transport there, so active transport of pyruvate in ADP, and talked about how active transport is actually involving an energy transfer from ATP, because then that would be another thing I could have mentioned there, a little bit more detail, and also that covers another topic. Final paragraph. 2 important uses of ATP in cells are nervous signaling and muscle contraction, which are vital for organisms to be able to move and respond to their environment, but also muscle contraction of the heart is necessary to produce the force to move blood around organisms to deliver oxygen and dissolve substances to all cells in the body. So this is kind of an important sentence. I've not gone into detail and I've not used these to talk about energy transfer. What I have said here is linking this idea of nervous signaling and muscle contraction to importance. I'm going to go and talk about muscle contraction, not nervous signaling. I don't think I'd have enough time to talk about nervous signaling in detail as well, but you could obviously include that if you had more time. But I would say I've just put that as a bit of an importance question at the start this time rather than at the end. If an electrical impulse arrives at a neuromuscular junction, the sarcolemma is depolarized, which causes an influx of calcium ions. Calcium ions diffuse into myofibrils from the sarcoplasmic reticulum. They bind to and cause tropomyosin to move, exposing the binding sites on the actin. Myosin heads attach to binding sites on actin, forming an actin-myosin crossbridge. Energy stored in the myosin head transferred by ATP hydrolysis is transferred to kinetic energy as the myosin head bends, which pulls the actin filament along in the power stroke, shortening the sarcomere and contracting the muscle. A new ATP molecule attaches to the myosin head, causing it to detach from the actin binding site. Hydrolysis of ATP to ADP and inorganic phosphate by ATPase enzyme transfers the energy needed to recock the myosin head to its original position, ready to form a new bridge, a cross bridge at the next binding site. Then I go into my importance finally about this specific example. Without the binding of ATP, the actin-myosin cross-bridge would not be broken, and the muscle would remain in its contracted state, and the energy from the power stroke, which contracts the muscle fibers, would not be transferred to the myosin head. So I've got my energy transfer from ATP importance in muscle contraction in as well at the end of this paragraph. So you could then do a sum up if you wanted to, but you should have signposted enough as you went along with your essay that you should not need to do a sum up saying, oh, this essay has clearly shown that there are. You could do that first paragraph at the end if you wanted to, the little kind of sum up at the end instead, but we've covered plenty of topics. We've talked about detail. Again, this muscle contraction here is all A-level detail, that's correct. Same with the detail that we gave in the respiration paragraph. And it's linked, most importantly, we linked this like energy store, transferring of energy to kinetic energy, to the movement of the myosin heads. That is clearly making links to the question as well. So that is the essay that I've written for energy transfers. Now let's have a look at like what other topics we could have maybe included and some tips on what to do and what not to do when you do your essay, and maybe to help you mark yours as well. Okay, so they don't give a mark scheme any more specific than the general mark scheme that we've already looked at. In the mark scheme for these questions, they will give you the topic list, which is not exclusive, so there are other things you may be able to include, but this is for the examiners when they mark your essay. They'll put these little mark codes next to where you've talked about the various spec points, so they can easily see, have you covered 4 topics at a minimum? So we obviously talked about photosynthesis, we talked about energy transfers through ecosystems, we talked about respiration, we talked about ATP, we also talked about muscle contraction. So we've got at least 4, if not 5, spec points covered here. There are other things you could have said, obviously, and they're listed here. So you could have talked about absorption, so that's like active transport, basically, because you'd have to talk about energy. You couldn't talk about diffusion because it doesn't require energy. Mass transport we touched briefly on with this idea of ATP being used in muscle contraction for the heart, but also if you think about active loading in plant cells, and that's used for mass flow, that would be a lovely one to talk about here as well. Stimuli responses and nervous impulses again, so anything where you've got the use of the sodium-potassium pump, which you can talk about for nervous impulses and stimuli responses, and that idea of energy being used and energy transfer. I think we've focused on the topics that are easiest here to relate to the question, and that's the thing. So what to avoid, this is taken from examiner reports that have marked whole cohorts of essays similar to this title, and these are kind of like the things that they stood out as the weak areas. So these are the things to avoid. So if you've done any of this, or you think you've done any of this in your essay, this is what kind of need to look out for to try and improve. So not focusing on the transfer of energy, because that is what is in the question. So describing energy, describing how ATP is made, but not, you could talk about all the things we talked about without using that phrase transfer of energy if you wanted to. But we made it very clear that we were talking about energy transfer when we were discussing our points. Not including both the transfer of energy within between the organisms, so not covering the whole question. The between organisms part really is limited to that idea of food chain. So if you avoided that, you wouldn't be answering the question. Talking about transfer of energy from organisms to their environment, so heat loss or something like that, that's irrelevant. It's not talking about the energy transfer between organisms and the environment. It was just inside organisms and between organisms. Some students made the mistake of covering all of respiration and photosynthesis, so just saying, here's everything I know and here's all the detail about all the parts of both, not being selective about where energy is being transferred and stored, mentioning energy stored in bonds and things like that, and being really specific about the bits of those processes that you're going to talk about to make sure you can focus on energy transfer. Spending too much time talking about ATP formation and not how the energy transferred by ATP is used in processes with examples. Not focusing on feeding relationships or focusing on feeding relationships without any reference to energy. So talking about predator-prey relationships or something like that. No, not relevant. Talking about nutrient cycles without linking energy transfers. This is really hard to do. So actually talking about energy transfers and the cycles, the nutrient cycles, and linking it to energy transfer is actually really, really difficult. So some people just kind of wrote all about energy and nutrient cycles, but didn't have any link to energy there. Okay. So hopefully that was helpful, and hopefully you've now kind of got an idea of what a model answer for those essays would look like. Obviously there's topics that I didn't include. If you want any help with writing the essay specifically, we have an essay workbook on the website that you can download that is based around kind of loads of different examples of essays and examples of content that you can include in different essay topics. I'd definitely say go and check it out. Have a go at writing the essay, then look and see what our model essay was and how it was marked and the examiner's comments. And you can pick out these kind of common errors and common mistakes and just make sure that you aren't someone that does that. And hopefully this has helped you kind of plan what you're going to do.
Ouch! This is why in some videos I'll explain scratches.